Hello students, so in our series of crash course, today we'll study the day 37 of which we will cover biomolecules. So we have already covered a part of it in which we studied about carbohydrates and its type. So we studied about its two types, which is oligosaccharides and your monosaccharides. Today we will study about the polysaccharide and then we will move on to other biomolecules such as your amino acids, proteins and vitamins. So this is our disclaimer. As I said, today we are going to study about polysaccharides. So polysaccharides are those carbohydrates that release a greater number of saccharides upon hydrolysis. So they should be greater than 12. Next, we will study about protein. Then we will see the classification of proteins and the structural levels of protein. So your protein is built up in many levels. It is primary, secondary, tertiary and quaternary finally, which gives you the complete structure of your protein. So we'll study about how these structures are built, what are the interactions that are responsible for the build up of the structure and how they can be formed and specific for a given function. Okay, so different proteins have different functions. Then we'll study about enzymes. So enzymes are also proteins that speed up biological reactions. Then we'll study about vitamins. After that, we will move on to nucleic acids, which are responsible for storing our genetic information. Lastly, we will discuss the biological function of nucleic acid. Okay. So first of all, what are polysaccharides? Now, carbohydrates that produce large number of monosaccharide units upon hydrolysis are called polysaccharides. Some examples of polysaccharides are starch, cellulose and glycogen. So starch is and your cellulose is mainly found in plants and glycogen is found in the animal. Then remember all your polysaccharides, irrespective of the type you are taking, they all are non-reducing sugar because they don't have a free CHO or carbonyl group. Remember, this is a very important point about the polysaccharides. For the case of your monosaccharides, every monosaccharide is possible of giving the reducing test. But for the oligosaccharides, I would specifically say for it the disaccharides, sucrose were the one which were non-reducing. But here, all the polysaccharides are non-reducing. Then polysaccharides are not sweet in taste, so they are considered under non-sugars, which is why they are also known as non-sugars. Now other saccharides, if you consider, such as sucrose and lactose, they have a sweet taste and they are responsible for the sweetness of your milk. So for example, lactose is present in milk, then fructose is present in fruits and all, and they are the one which gives them the sweet taste. But in case of polysaccharides, they don't have a sweetness in their taste and they are considered as non-sugars. Now let us continue on the discussion of your polysaccharides. So some important polysaccharides are mentioned here. First is starch. The main storage polysaccharides of plants is starch. It acts as a primary food reserve to fuel growth and development. It is a polymer of alpha D glucose unit. So you will always remember this, that they are formed from the alpha D glucose. Now it has two components. The first one is amylose, which is soluble in water, and the other one is amylopectin, which is insoluble in water. Then moving on to the next polysaccharide, which is cellulose. So your cellulose is mainly found in the cell wall of the plant cell. It is the main component of the cell wall. It is a straight chain polysaccharide made up of beta D glucose units and is linked by beta 1,4 glycosidic linkage. Beta 1,4 glycosidic linkage means C1 and C4 are joined by a O in the middle. This means a bond is formed between C1 carbon at the first position and one glucose, and C4 carbon means carbon at the fourth position of another glucose unit. and this is the way your cellulose is formed. So this is the basic difference between the cellulose and starch. Starch is made up of alpha D glucose, whereas your cellulose is made up of beta D glucose. Now, humans cannot digest cellulose because the cellulose enzyme is not present in our digestive system. So cellulase is a special type of enzyme which is responsible for digesting the cellulose. You will mainly see that the names of the enzymes are derived from the species which it can act upon and decompose. For example, maltase. So maltase acts upon the maltose. Similarly, here we have seen cellulase which acts upon the cellulose. Now continuing our discussion, the next thing that we will study about the polysaccharides is the glycogen. So this is the third type of polysaccharide which is important for your JEE exams. The primary storage form of carbohydrates in animal and including human is glycogen. It is also called as animal starch. It is present in liver, muscles and brain. Glycogen is a condensation polymer of alpha D glucose. Okay, so it is also a polymer of alpha D glucose. Next, when an animal or human needs a quick boost of energy, such as during exercise or fasting, enzymes rapidly break down the stored glycogen into glucose, which is then released into the bloodstream from the liver or used directly by muscle cells as fuel. Now, the next topic is protein. So, we studied about three types of polysaccharide: starch, cellulose and glycogen. So, always remember the monosaccharide unit which is responsible for the build of these three polysaccharides. Our next topic is protein. So proteins are large biomolecules which are found in every living organism. Protein shows Burette test, Millen's test and Ninhydrin test. Then they are the polymer of amino acids. Next, the total number of amino acids found naturally in protein is 20. Okay, so this is the minimum number of amino acids which should be there to form a protein. So first let us understand what is amino acid to understand the importance of protein. So amino acids are compounds that contain both acids COOH and amino group means NH2. Proteins are polymer of amino acid, meaning that when many amino acids are joined together, a protein is formed. So amino acids, as the name suggests, contain both amino and acid. This means that they have both amino and acids. However, the behavior of amino acids is not like any simple amines or carboxylic acid. They behave like salts, not like amines or acids. Amino acids have both acidic and basic group in the same molecule. Due to this, they do not react like acid or base, but rather they behave like salts. Okay, so here is a diagram in which RCHNH2 C double bond OOH is an amino acid which is interconvertible into RCHNH3 positive C double bond OO negative. Okay, so you can completely see that how your previous structure is getting transformed into a Zwitter ion. So whenever you see a positive and a negative charge, both are present on the same molecule, you call that molecule as Zwitter ion. Here you can see that how your carboxylic acid or the COOH group is transferring its H to the NH2 and creating a salt within the molecule, in which the positive part is NH3 plus and the negative part is COO minus. Now, at a certain pH of particular medium, the structure behaves like a dipolar ion, also called as the deuterium. This pH is called as the isoelectronic point of amino acids. So there is a certain pH and it is different for different types of amino acids. So multiple amino acids are possible, including your essential and non-essential, and they have different isoelectronic point depending upon their structure. Got it? When we pass current through it, it does not move towards any electrode because of the balanced charge, right? It contains both the positive and negative charge. It is also called dipolar for this reason, because it has both negative and positive charges. So it is very obvious, although this is containing charge upon it, but due to the balance of charge, it will neither be attracted towards the anode nor towards the cathode. Got it? Now moving on to the classification of amino acids. Based on their synthesis, amino acids are divided into two classes. First is essential amino acids. What are essential amino acids? These are amino acids that the body cannot synthesize on its own. So, we have to obtain them from our diet. Now, examples of essential amino acids are valine, leucine, lysine, isoleucine, arginine, etc. The second class is your non-essential amino acids, which are produced in our body. So, it's very simple. If it is not synthesized in our body, then we have to take them externally from any food source, right? But if it is synthesized in our body, then we do not need to take it as the food source. Therefore, they are named as essential for us and non-essential for us based on our consumption. These amino acids can be synthesized inside the body, and examples of non-essential amino acids are glycine, alanine, glutamic acid, and aspartic acid. Then, how does a protein is formed? So, it is formed due to the formation of peptides and peptide linkage. The basic structural unit of protein is alpha amino acid. An alpha amino acid is an organic molecule containing both an amino group NH2 and a carboxylic group COOH on the same carbon atom, known as the alpha carbon. Amino acids can be joined through an amide bond. When amino acids join together, the amide bond between them is called a peptide linkage. This bond is formed between the carboxyl group COOH of one amino acid and the amino group of another amino acid. This process is called condensation reaction because one molecule of water is released. So peptides are short chains formed by the joining of amino acids and a peptide bond. And a peptide bond is the chemical bond that joins those amino acids together. So let us see an example on the same. When we react amino acid such as NH2CH2COOH, so this is your glycine. This is the only optically inactive amino acid. Okay. All other amino acids are optically active. Only glycine is optically inactive because here the only one carbon that is present has two same groups, which is hydrogen. What is the other amino acid that we are taking? It is NH2CH3COOH. And when we perform dehydration, an H2O molecule was released from this reaction. The COOH of one amino acid and the NH2 of another, these two are joined together through a peptide linkage. Therefore, we got a peptide linkage between the molecule, which is CONH. Here O is doubly bonded to carbon, and we got a kind of peptide molecule which is dipeptide, having formula NH2CH2CONH CH CH3 COOH. So here only one water molecule is being removed from here. Got it? Now, moving on to the next thing. So if we talk more about this, when two amino acids are joined together by a single peptide linkage, the molecule formed is a dipeptide. So it's very obvious. When three amino acids are joined together, the molecule will be named as tripeptide. And if 2 to 10 amino acids are present in a peptide, they are called as oligopeptide. Similar to the case of saccharides, like your oligosaccharides, we have oligosaccharides, we have this oligopeptides, where if there are more than 10 amino acids residues, those peptides are called polypeptides. Poly means many. So if there are more than 10, they are known as polypeptides. A polypeptides with a molecular weight greater than 10,000 U, unified mass, is called a protein. Okay. So in this way, your amino acid is forming your protein. So first you have your amino acid, right? Now, your amino acids are combining with each other with the help of peptide bond, and they are forming longer and longer chain. If your amino acid count is between 2 to 10, we name them as oligosaccharides. If it is greater than 10, we name it as polysaccharide. Further, we are taking a particular mass. So basically, if the overall atomic mass, if we add them together and we see that the molecular mass is now greater than or equals to 10,000 U, then we name that specific structure as protein. Okay. So how can you define protein in a structural sense? It is basically a condensed product of many amino acids which are joined together by peptide linkage and have a molecular mass greater than 10,000 U. Got it? Okay. Now moving on to the next part. The next topic is classification of protein. So, your proteins can be divided into two types based on their molecular shape. The first one is fibrous protein. So, those polypeptide chain which combine to form a fiber-like structure as seen in keratin and myosin. In keratin and myosin, polypeptide chains are in a fibrous structure. They have long threads like polypeptide chain that run parallel to each other, forming sheet-like structure. Got it? These are usually insoluble in water. The next type is globular protein. These structures are formed when polypeptide chain coil around each other and form a spherical shape. These are usually soluble in water, such as insulin and albumin. Insulin and albumin are two examples of globular protein, while your keratin and myosin are the two examples of fibrous protein. So you should slowly feed all this data in your mind, and they will be directly asked in your exam. So what did I say in this whole slide? First of all, your protein is divided on the basis of their molecular shape. So in case of your fibrous protein, they form a sheet-like structure. Okay. Now in that we have keratin and myosin. The other one is a globular protein. So basically they coil together and form a spherical shape. Got it? And in that we have the insulin and albumin. Your fibrous protein are what? They are insoluble in water, whereas your globular protein are soluble in water. Okay, so remember these points in your mind. Next, moving on to the structural levels of protein. So I would say that this is the most important part from the protein thing. Your structure and shape of protein have 4 different levels. Let us understand them one by one. Okay, so first of all, the primary structure. What do we mean by primary structure of protein? So they involve the primary structure of a protein involves a polypeptide in which amino acids are joined in a specific sequence. If we change the primary structure, we get a new protein. This was about the primary structure. So in primary structure, we are mainly interested in the amino acids that are added. Okay, the first step. Okay, so basically they will be added together in the form of your what? In the form of a polypeptide chain. You got your linear structure, and this represents the primary structure. Next is secondary structure. So polypeptide chains are joined together by hydrogen bonds. These hydrogen bonds occur between the carbonyl and the NH group of the polypeptide due to the folding of polypeptide backbone. Now in this we have two types. One is alpha helix and the other one is beta plated sheet. So there is a sheet structure and a helix structure. So what is being told here? So there can be two scenarios. The first scenario is that in the alpha helix case, two loops can be joined together, like they can be curled around together to form a linear chain. Or the second type is known as the sheets. So in that case, your one single strand can be like going on in a wavy pattern, and now they will be connected together. Okay, in the intermolecular forces will act, and that intermolecular force will be your hydrogen bonding, and hence your sheets are formed. So two types you should remember. One is helix and the other one is beta plated sheet. Got it? After that, what will happen? So once two coils are joined together, we will move to the tertiary structure of protein. It represents the overall folding of polypeptide chain. So basically, now the strands are joined together. Now they will fold. Now multiple strands will be folded together. It represents the overall folding of polypeptide chain. It includes polypeptide bond, hydrogen bond, disulfide link, van der Waals force, and the electrostatic force. So in secondary structure, only hydrogen bonds are formed. In the tertiary structure, we have hydrogen bond, disulfide link, van der Waals force, and the electrostatic force. These forms two main molecular shapes, fibrous and globular. So from this part only, we are getting two types of protein. It is basically divided due to the tertiary structure, difference in the tertiary structure. Now the last one is the quaternary structure. This structure is formed by two or more polypeptide chains which are classified as subunits. These subunits are spatially arranged in that is 3D arranged with respect to each other. Got it? So there are 4 layers. The first layer is what? Joining of amino acids to form a linear chain. Got it? The second is joining them together. Okay, means coiling them together or either making a sheet together by hydrogen bonding. Got it? Next is bending of that sheet. Okay, now that one sheet is formed or either a coil is formed, two possibilities are there. Now they will bend to form the tertiary structure, which are stabilized by disulfide bond, hydrogen bond, then your van der Waals force and the electrostatic force. In this way, many structures would be formed. So they can be fibrous or globular. Now they will come together. Such structures will come together and finally form the quaternary structure. Got it? So you can take the example of hemoglobin. Okay. In the case of hemoglobin, there are 4 subunits. They just come together and form the quaternary structure. Moving on to the next, which is denaturation of protein. So it is basically destructing the structure of protein. How it is done? So during denaturation, only the secondary and tertiary structure of proteins are destroyed. Remember, this is very important to remember. The primary structure is remain unaffected with no changes in their linkage. Okay, means the peptide linkage are still there, but the hydrogen bonding and the disulfide bond all are being broken. So such process is known as denaturation. When there is a disturbance in hydrogen bond due to acid, heat or alcohol, so there can be many external factors. So acid means changing pH, heat changing the temperature, and alcohol changing the reaction. Okay, then what will happen? It will result in the unfolding of globules. Similarly, the helix can also uncoil. So you know it, helix means the coiling of 2 strands together to form a linear chain, but they can be uncoiled again due to the change in temperature or change in pH. And in this way, your protein loses its biological activity which it was performing. Got it? So this is the reason you would see that whenever your body is experiencing a small heat change also, so you know there is a specific temperature at which your body should be consistent. If there is a small change in your temperature, then the functioning of your protein is disturbed. As a result of that, you get sick and all, right? So this is the main reason behind disturbance of your health through temperature change. Similarly, if your pH has been changed of your blood or of your stomach, then that would also result in loosening of the function of your protein and hence disturbing your health. Got it? Now our next topic is enzyme. What are enzymes? So almost all enzymes are globular proteins. So they are just a type of protein, but the globular one. Now they act as biological catalysts, which means they speed up reactions without being consumed themselves. Now these are very specific for a particular reaction or a particular substrate. Each enzyme has a uniquely shaped active site that is complementary to only one specific substrate molecule, much like a key fits into only one lock. This precise fit ensures that each enzyme only catalyzes a particular reaction, preventing unwanted side reactions and bringing order and efficiency to the thousands of metabolic processes in a cell. Okay. Now, next is enzyme inhibitors. Reduce the activity of a particular enzyme. These are mostly inorganic ions or complex organic molecules. When there is a deficiency of hydrolysis or trisinase enzyme or congenital disease like phenylalbinism can occur respectively. So these are some specific examples you can see here, which is formed due to the lack of enzymes. Now moving on to the vitamins. So vitamins are organic compounds which are necessary in small amounts in our diets, which means their quantity should be very low for our normal growth and to fulfill the specific nutrients required for our health. So nutritional supplements that are needed in small amount in food to maintain normal growth and health are called vitamins. Provitamins are biologically inactive compounds whose structure is similar to vitamins and they are easily converted into active vitamins. For example, Beta carotene is a provitamin of vitamin A. This means if we convert beta carotene into its active form, it will act like vitamin A. That is, the structure of beta carotene and vitamin A are quite similar to each other. Next, we will see different type of vitamins. So, first of all, we have fat or oil soluble vitamins such as vitamin A, vitamin D, vitamin E and vitamin K. The other type is water soluble such as B group vitamins like vitamin B, vitamin B12, B11, etc. and vitamin C. So you must remember this always. What are the fat soluble vitamins are A, D, E and K, whereas water soluble are B and C. So obviously this is coming due to their polar nature or hydrogen bonding. That is the reason their solubility is changing. Now continuing further with the vitamins, now we will look at a table 2.3 which describes some important vitamins, their sources and problems caused by their deficiency. The first vitamin is vitamin A, also called retinol. Its sources are fish oil, liver oil, carrots, butter and milk. We get vitamin A from all these things. If there is a deficiency of vitamin A, it can cause xerophthalmia, meaning the cornea of the eye hardens, or blindness can also occur, such as night blindness. The second vitamin is vitamin B1, also known as thiamine. It is abundant in yeast milk and green vegetable. If there is a deficiency of vitamin B1, what disease can occur? It can cause beriberi, which results in loss of appetite. The third vitamin is B2. It is also called riboflavin. Its sources are milk, egg white, liver and kidney. If there is a deficiency of it, what will happen? Cirrhosis will occur. When the corners of the mouth and the lips dry up, that condition is known as cirrhosis. The fourth vitamin is B6, also called pyridoxine. Its sources are yeast, milk, egg white and grains. The diseases caused by its deficiency are convulsions and nervousness. Okay. Now moving further. So here is a table which will describe about important vitamin, their sources and problems caused by their deficiency. So the fifth one is vitamin B12, also known as cyanocobalamin. Okay, it is mainly found in meat, fish, egg and curd. If there is a deficiency of it, the disease that occur is called pernicious anemia, which is actually caused by the deficiency of RBCs in hemoglobin. The sixth vitamin is vitamin C, also called ascorbic acid. Its sources are citrus fruit, amla and green leafy vegetable. If there is a deficiency of vitamin C, the disease scurvy occurs, in which there is a bleeding from the gums. If gums are bleeding, one may have scurvy. The seventh vitamin is vitamin D. Its source is exposure to sunlight. And food sources are fish and egg yolk. And if there is a deficiency of vitamin D, then diseases like rickets and osteomalacia can occur. The next vitamin is vitamin E. Its sources are wheat, germ oil, sunflower oil, etc. Its deficiency increases the fragility of RBCs and can lead to muscle weakness. And the last one is vitamin K, which we get from green leafy vegetables. If there is a deficiency of vitamin K, blood takes longer to clot. Okay. Now we will move on to the next important topic, which is recently asked in your exams, that is nucleic acids. They act as a chemical carrier of genetic information in cells. Genetic information of cells is chemically present in nucleic acid, which is further transferred. Nucleic acids are responsible for protein biosynthesis. Okay, so basically this genetic material carry the mechanism for synthesizing a proper protein, and with the help of that genetic material, your nucleic acids help in the biosynthesis of protein. Now, nucleic acids are biological polymers of nucleotides. So, proteins are polymers of amino acids, whereas your nucleic acids are polymer of nucleotides. A nucleotide contains a deoxyribose, sugar, phosphate and a nitrogenous base. Now, there are multiple nitrogenous bases. A nucleoside contains deoxyribose and a nitrogenous base. So the only difference between a nucleoside and a nucleotide is that the nucleotide has a phosphate attached to it, which will help in developing phosphodiester bond. Got it? Whereas in nucleoside, we have only deoxyribose sugar and the nitrogenous base. Got it? A nucleoside is formed when the base attached to the first position of the sugar. Now, what about nucleotide? In nucleotide, the carbon of the sugar are numbered as 1 dash, 2 dash, 3 dash, so that they can be distinguished from the carbon of the base. After that, when the fifth position of the nucleoside sugar moiety joins with a phosphoric acid, we get a nucleotide. This concept is very important for the basic definition of your nucleoside and the nucleotide. You will always remember what are the components of nucleosides and nucleotides. So the nucleoside has only deoxyribose sugar and nitrogenous base, whereas your nucleotide has deoxyribose sugar, phosphate and nitrogenous base. Why is this phosphate present here? So it is responsible for the polymerization process. I said that your nucleic acids are formed by the polymerization of multiple nucleotides, and that bond, or I would say that bonding between your two nucleotides is done with the help of this phosphate. Okay. So nucleotides are joined together to each other by a phosphodiester bond, which is formed between the fifth and the third carbon atom of the pentose sugar. Then moving on to the types of nucleic acid. So there are mainly two types of nucleic acids. The first one is known as deoxyribonucleic acid. What are deoxyribonucleic acid? So they are popularly known as DNA. So it is a polymer of nucleotide. And what does a nucleotide contain? Deoxyribose sugar, phosphate and a nitrogenous base. Got it? It is genetic material and genetic information is stored in our DNA. It has a double helical structure made up of two helical strips. Okay, similar to the case of alpha helix. In the case of protein, your DNA is formed by a alpha helix structure, but here the components of the alpha helix are nucleotides and not your amino acids. The four bases which are there in your DNA are adenine, guanine, cytosine and thymine. They are having a code for each of them. For adenine it is A, for guanine it is G, for cytosine it is C and for thymine it is T. Adenine and guanine are called purines, while the thymine and the cytosine are known as pyrimidines. Remember, this is very important to remember. Now, next is the hydrogen bonding. I told you earlier that two coils are moving in a alpha helix form. But what is the force that is keeping them together? That force is your hydrogen bonding. So, between two bases, which is your adenine and thymine, there are two hydrogen bonds. Okay, remember, this is very, very important, and this can be asked in your exams as well. Whereas between the two bases, which is your cytosine and guanine, three hydrogen bonds are formed. Got it? So whenever you see in one strand, if you are seeing A, then the next corresponding part that would be present, it will be T. Okay, and if one strand is containing G, then the next corresponding part would be C, and the opposite is also true. Got it? Suppose the sequence of a DNA goes like this: A, G and T. So for the next corresponding strand, what will be the sequence? So I said it, A, G and T. So the next one will have, instead of A, it will have T. Got it? Instead of G, it will have C, and instead of T, it will have A. So they are complementary to each other. Remember, this is very, very important. Now moving on further, the next type of nucleic acid is RNA, also known as ribonucleic acid. Now, it is also a polymer of nucleotide, but in nucleotide unit, ribose sugar is present instead of deoxyribose sugar. Got it? And what are the bases that are present? The bases are adenine, guanine, cytosine and uracil. So uracil is present in RNA instead of thymine, which was present in DNA. So this is the difference between your RNA and DNA. Next, there are three types of RNA. First one is mRNA or also known as messenger RNA. Second is rRNA or known as ribosomal RNA. The third one is tRNA or transfer RNA. They perform different functions. mRNA carries genetic information to the ribosome, and rRNA forms the ribosome structure, catalyzing the formation of peptide bonds to build proteins. All three work together in the vital process of protein synthesis. Okay. Now we will try to compare RNA and your DNA. So what are the main points of differences between your RNA and DNA? We will discuss this, and this can be asked in your exams as well. So the first one is that your DNA has a ability to do self duplication. Okay, so whenever a cell is divided, it has the capability to do the what? Self duplication. Then identical DNA strands are transferred to the DNA to the daughter cell. In this way, DNA is responsible for maintaining the identity of different species of organism over millions of years, and your genetic information is being transferred from your forefathers to you in this way with the help of the DNA. Sorry for the disturbance. Now, next, the RNA molecule synthesizes different type of protein inside the cell, but the messenger RNA, okay, the message for the particular type of protein is present in the in DNA. If we want to synthesize a protein, its message will be in DNA, but the RNA molecule performs the synthesis. Okay, so that is the duty of your RNA. Genetic messages are encoded in DNA, while RNA translates the message encoded from the DNA and helps in making protein. In this way, RNA performs the protein synthesis. So this was all for the theory part. Now we will do questions on it. Just wait for a while. I will open the questions. So here is the first question. Among the pairs given in the options, we have to choose which pair gives a positive tolerance test. Options are glucose and sucrose, second, glucose and fructose, third, hexanol and acetophenone, and the fourth one, fructose and sucrose. So you might just give your answer in the comment section. I will just wait for a while. Then we will move on to the correct answer and then the solution part. I will wait for a minute. This is direct theoretical question. Remember, since you have to look for the positive tolerance test, then only aldehydes are capable of showing a positive tolerance test. Okay, and the primary alcohols are also capable of showing a positive what tolerance test. At the same time, in the case of carbohydrates, if your C1 position means the hydroxyl present at the C1 position, if it is free, then it can also give tolerance test. So what will be the correct answer for this question? So I will wait for 15 more seconds. Then we will move on to the correct answer. So your time is up. Now let us discuss the option one by one. First is glucose and sucrose. So when we were studying in the theory part, we already seen that all the disaccharides except your sucrose is a non-reducing is a reducing sugar. Since sucrose is there in the first option, so it is a non-reducing sugar, and hence it will not give positive tolerance test. The next option is glucose and fructose. So both of them have a free aldehydic group. Okay, both glucose and fructose have a free aldehydic group. But the problem is that with the option C. Okay, so moving on to option C, what is written? It is hexanol and acetophenone. So hexanol is having an aldehydic group, of course, but your acetophenone is having a ketonic group. Then the last one is your fructose and sucrose. Again, your sucrose is there in the option D. So option A and D are already eliminated, and C is also eliminated. So we are only left with the option B. Hence, the correct answer would be option B. So glucose and fructose both can give the positive tolerance test. Now let us move on to the solution. So it's very simple. Glucose and fructose gives a positive tolerance test because they have a free aldehydic group present, which is why both are reducing sugar. Tolerance test is used to identify the aldehyde group, and sugars that have an aldehyde group present gives us the positive tolerance test. Therefore, the both correct option would be option B. Moving on to the next question. Beta and alpha glucose have different specific rotations. When either of them is dissolved in water, their rotation continuously changes until a single value is obtained. What do we call this process? Options are epimerization, resmization, anomerization, mutarotation. Okay, you have to choose the correct option. Now, what will be the correct answer? It's a direct one, so I will give you 1 minute to think about it and give me the correct option. Then we'll move on to the correct answer and the solution. Okay, so I would say, tell you about the previous question, one important thing. So I said there that your fructose is capable of giving a tolerance test, but it has a ketonic group. So how it can be the correct answer? So it can isomerize. I would say it can isomerize into a aldose, right? Because this whole tolerance test is done in the presence of your base, and that base is capable of isomerizing your fructose into a free aldehydic group, and hence it can give your tolerance test. Yes, the correct option word for this question would be what? So it's very obvious, it would be mutarotation, which is option D. So it's not option C, it's your option D. Now your beta and alpha glucose are two types of glucose, and their specific rotations are different. We have seen that when either of them is dissolved in water, their rotation keeps changing until a single value is fixed. and that value is plus 52.5 degrees. This process is called as mutarotation, and hence the correct option would be option D. Moving on to the next question. We have to tell which statement about glucose is not correct. Options are: it is an aldohexose. Second, when it is heated with Hi, it forms N-hexane. Third is, it is present in furanose form. And fourth is, it does not give 2,4-DNP test or 2,4-dinitrophenol test. Out of these four options, which one will be the correct one? Which will be the incorrect one about glucose? You have to choose that. So I am giving you 2 minutes to think about it. Then we will move on to the solution. So we have talked a lot about glucose in the yesterday's class, and it is quite clear that what will be the correct answer. Think about the structure and think about the reactions that we have. And there is a statement related to the furanose form. Remember that your furan and pyrene, two were the structures with which we were comparing our aldehyde and fructose. Okay, and one of them resembled your furan and one of them resembled your pyrene. So from there you will get your answer. Now before moving on to the solution, we will try to test each options one by one. First of all, it is an aldohexose. Here it is, yes, it is true. It has an aldehydic group and it has 6 carbons. So it will be an aldohexose. Second one, it reacts with Hi to form an hexane. Okay. So since it has 6 carbons and each of them is containing oxygen on it, so when we reduce it with Hi, which is a reducing agent, it will reduce this whole glucose to form a linear and simple structure like N-hexane. Third one, it is present in furanose form. So this one is the incorrect option. Yes, option C will be the incorrect because it is present in the pyranose form. Fourth, it does not give 2,4-DNP test. So this is also correct. Now, the correct option would be third one, which is a wrong statement for the glucose. So the cyclic structure of glucose is shown as a pyranose ring in the Haworth projection, not as a furanose ring. And the pyranose ring is used to represent your glucose, whereas your furanose is used to represent your what? Fructose. Got it? So you can also remember it. So since they both are starting with F, your furanose and your fructose, you can remember with this small trick. So glucose is present in the pyranose form in the cyclic structure. The word pyranose means it has a 6-membered cyclic ether where one of the member is oxygen. And what about your next one, furanose? So in that we have 5-membered ring in which one is oxygen. Okay, so furan gives your furanose and pyrene gives your pyranose. Therefore option C will be the correct one. Moving on to the next question. Invert sugar is a equimolar mixture of which of the following compound? Options are: first, D-glucose and D-galactose. Second, D-glucose and D-fructose. Third, D-glucose and D-mannose. Or fourth, D-glucose or D-ribose. So invert sugar, the term which is being referred is for a special type of carbohydrate, and it is defined for that carbohydrate because upon hydrolysis it gives opposite signs. Okay, so that is the reason, opposite signs of rotation. So that is the reason we call it as invert sugar. Now you have to tell that what are the components of that invert sugar. So it's a easy one if you remember the name. So we have option coming as B. So anyone else who is interested to give the answer for this question? It's a easy one. So you can see here that in your JEE exams also theoretical questions are asked directly from your biomolecules, and hence you can easily score number in this chapter. Got it? So I will wait for 1 more minute to give the answer for this question. So invert sugar is referred to. So let me give you a hint. Invert sugar is referred to sucrose. Now you have to only tell that what are the components of sucrose or what we will get upon hydrolysis of sucrose. Okay, so we have one more answer is option B. Anyone else who is willing to give the answer? So 30 more seconds. Remember, invert sugar is a term which is often used for sucrose because it gives us opposite sense of rotations upon hydrolysis from the parent one, and it is composed of two units. Now can you tell me what are the names of this D-glucose and D-fructose? Okay, so D-glucose and D-fructose is the correct answer, but whether it is alpha or beta, that can also be a good question. So you know that the D-glucose is present in the alpha form and the fructose is present in the beta form. So D plus sucrose, which is an inverted sugar, is an equimolar mixture of D plus glucose and D minus fructose. So equimolar mixture of D plus glucose and D minus fructose together form the D plus sucrose. D plus sucrose is called invert sugar. Why? Because sucrose is dextrorotatory. When it undergoes hydrolysis, it produces D glucose, which is dextrorotatory, and D fructose, which is strongly levorotatory. Since the lever rotation of fructose outweighs the dextro rotation of glucose, the overall optical rotation of sucrose is inverted. This is why we call it as invert sugar, because the sign of rotation is inverted and now it is levorotatory in nature instead of its parent molecule, which was dextrorotatory in nature. Hence, the correct option would be option B. And what are the names of your glucose and fructose? So glucose is of the form alpha, whereas fructose is of the form beta. Moving on to the next question, it is asked that among the given compounds, which compound does not give Burette test? Options are carbohydrates, polypeptides, urea, or proteins. So we have already talked about this in the theory part, that there are a certain type of biomolecules which are capable of giving us the Burette test. So what will be the correct answer according to that? So 1 more minute is left. Just write your answer in the comment section. Okay, so your time is up. So let me give you a brief overview about the Burette test. So it basically involves a complex ion of Cu2 plus, and greater is the number of your peptide bond, greater will be the formation of a complex with the help of the nitrogen of the peptide bond. You know it very well that peptide bond is formed by the condensation reaction of your COOH group of one of the amino acid and NH2 of the other amino acid, and as a result we get an amide. So basically the nitrogen of your amide, okay, it forms a complex upon processing it under the Burette test. Got it? And as a result of that, a violet colored complex is formed. So greater will be the number of this peptide bond, greater will be higher will be the intensity of your violet color. Now according to that, what will be the correct answer? You know it very well that both your polypeptides and proteins will contain your amino acid and amino acid bond, which is your polypeptide linkage. Why? Because polypeptide is made up of a number of amino acids and that number should be greater than 12. And for proteins, it is also made up of amino acids, but their molecular mass should be greater than 10,000. But beside that, we have a peptide bond, so it will give the Burette test. Similarly, your ammonia also has an amide group present in it. If you remember the structure, it has an amide group present in it. So it will also give the Burette test. So there is only one option which is being left, and it has no amide group present in it, and hence it will not give the burette test. Therefore, the correct option would be option A. Now moving on to the solution, as I said, compound that have a peptide bond, okay, NHCO, they give us the burette test. Carbohydrates like sugar do not have a peptide bond, therefore they do not show burette test. Hence our correct option would be A. Moving on to the next question, question number 14. So it is asked that which of these statement is correct. The first statement is all amino acids are optically active except lysine. Second, all amino acids are optically active. Third, all amino acids are optically active except glycine. And the last statement is all amino acids are optically active except glutamic acid. Now, if you have seen the structure of your amino acids, you will see that only one amino acid is present which is optically inactive. Got it? So you have to identify that one. Of course, option B will not be correct one. Now you are left with option A, C and D. Just write the correct answer in the comment section. Again, you can see that this is direct theoretical question. If you know the answer, you can easily get your marks in your exams. Remember, what are optically active compound? Those which contain more than one optical center and have no plane of symmetry are known as optically active. And what are optical centers? Those carbons which are joined to 4 different groups. Okay. Now from that point of view, can you tell me which will be the optically active compound and which one amino acid is optically inactive? So think about it and write your answer in the comment section. I am giving you 1 more minute. So already there we have an answer as C. Anyone else? Remember, all amino acids are alpha amino acids, or naturally amino acids are alpha amino acid. Means that NH2 and the COOH group are present on the same carbon. Okay. What will be the correct answer for this question? So, your time is up. The correct answer would be option C. All amino acids are optically active except your glycine, because it has only one carbon and that carbon is attached to 2 hydrogens. Got it? So moving on to the solution, the structure of glycine is NH2CH2COOH. So there is no chiral carbon present in it. You would say that this carbon, which is involved in the carbonyl formation, is also an optically active carbon because it has 3 different groups. But the problem is that this carbon that you are seeing, it forms a planar structure. Okay, it is sp2 hybridized. sp2 hybridized carbon are never optically active because they have a plane of symmetry. Got it? So there are certain criteria in which they can be optically active. So those are allenes and all. So there is no formation of allenes. Now we are left with the another carbon, means the CH2 carbon, but it is attached to 2 types of same group, which is hydrogen. The other 2 are different, NH2 and COOH, but the remaining 2 are same. So there are no chiral centers here, and hence this cannot be optically active. Okay. So apart from your glycine, no other amino acid is optically inactive. Got it? Now moving on to the next question. In this question, it is asked which bond plays the main role in the formation of secondary structure of protein. So we have talked about the primary, secondary, tertiary and quaternary structure. Options are A, coordinate bond, B, covalent bond, C, hydrogen bond and D, ionic bond. So what will be the correct answer? So in your secondary and tertiary structure, there were 2 kinds of interaction. Okay, so in the tertiary structure, we have disulfide interaction also, then hydrogen bonding also, and then electrostatic force also. But in the case of secondary structure, there was only one type of force. So what was that? You have to tell me. Okay. This covalent bond is only seen in case of the primary structure. Remember this. So now you are left with three options: coordinate, hydrogen, and your ionic. So what will be the correct answer? So we already have, so can anyone tell, just write it in the comment section. Okay, so we have an answer as C. Anyone else? So you have 1 minute to give the answer. Okay. So let us move on to the correct answer. It will be option C. So in the secondary structure of protein, the only force that is responsible for the built of the secondary structure is hydrogen bonding. Now moving on to the solution. The secondary structure of protein refers to the shape in which long polypeptide chain can exist. Now there are two types of secondary structure, we have seen it. One is alpha helix and the second one is beta plated sheets. Whereas the tertiary structure involves your globular and the fibrous. So hydrogen bonding determines the secondary structure of protein. Long peptide chains are held together in the secondary structure through hydrogen bonds. Hydrogen bonds are formed between the C double bond O and the NH group of the peptide bond. Alright. So in the question we are asked that what the secondary structure of protein is formed by, which means that it is determined by, and that is your hydrogen bond. Therefore the correct option would be option C. So also remember the type of hydrogen bonding. It is between the carbonyl and your NH. So it's very obvious because oxygen has a lone pair and in the NH your hydrogen is attached to an electronegative element. Moving on to the next question. Among the given compounds, which compound can be identified or detected by Molish test? Options are nitro compounds, sugars, amines, or primary alcohol. So what will be the correct answer? So let me tell you something about Molish test. So it is a qualitative biochemical assay that is used to detect presence of carbohydrates in a sample. Okay. So it is a universal test for all type of carbohydrates, monosaccharides or disaccharides or polysaccharides. Now what is the principle of the test? So this reaction is based on the dehydration of carbohydrates by a strong mineral acid, typically concentrated sulfuric acid. How does the dehydration occur? So sulfuric acid removes water from the carbohydrates, converting pentose into furfural and hexose into 5-hydroxymethyl-furfural. Now, in the condensation, this furfural derivative then reacts with alpha naphthol, which is the Molish reagent, to form a violet or purple colored complex. Okay, so this is your Molish test. So it's very clear on what will be the correct answer according to that. It is obviously your correct answer option would be B, which is sugars, because sugars are a type of carbohydrates. So this is basically coming from your analytical part. After this, we'll move on to the analytical. There you will know what is this Molish test, what is this Burette test, and what is Ninhydrin test. Okay. Now, carbohydrates, that is your sugar, can be detected by the Molish test. What do we do in it? So let us look at the steps involved in the Molish test. First, we need to put our test sample into a test tube with water and shake it so that it dissolves. After that, we add 2 to 3 drops of Molish reagent to the test tube and add drops of concentrated sulfuric acid along the side of the test tube. Now what will happen? Slowly purple colored rings will start to form. Got it? As soon as those rings start to form, it means that carbohydrates are present in our unknown compound. So this is our Molish test. We perform this test to detect carbohydrates. like sugar. So our correct answer is option B. Sugars are also, non-sugars are also carbohydrates, but since any sugar, any carbohydrate can give the smallest test, and so can your sugar. So with this we have covered the question as well. Now we'll do some DPPs regarding this chapter. So just wait for a while. I will open the DPPs. Here is the first question. So this question says that what is the primary chemical nature of enzymes? Options are A, carbohydrates, B, lipids, and then D, nucleic acid. Okay, C is globular protein. So what will be the correct answer for this question? Remember we talked about the enzymes. Enzymes is a form of proteins. Now you have to tell that which type of protein. There are two types of protein, right? On the basis of their structure, we have two types of protein. One is fibrous and the other one is globular. Since in the given options we only have the globular, we don't have to think much, but we know it for sure that the correct option would be option C, which is your globular protein. Moving on to the next question. So this says us that which of the following statements about enzymes is correct? First one, enzymes are consumed in the reaction they catalyze. So it is a very basic question. Second, enzymes lowers the activation energy of your reaction. Okay. Third one, enzymes are non-specific to their substrate, means it can act upon any substrate. Third, enzymes are primarily carbohydrates in nature. So we are sure about it that it is not consumed in the reaction. It is the basic definition of any enzyme that it does not participate in reaction but speeds up the rate of reaction. Then, and how does it speeds up the rate of reaction? By lowering the activation energy. So correct option would be option B. Moving on to the next one, the incorrect one. Enzymes are non-specific to their substrate. So this is particularly incorrect, because if I talk about catalyst, so enzyme is a type of biological catalyst, then catalyst can be acted upon any other substrate, but enzymes are very specific in nature. They will only fasten certain reactions in our body. Got it? What is the fourth one? So it is specific in nature. What is the fourth one? They are primarily carbohydrates. No, they are primarily proteins. So the correct option would be second one. Moving on to the next question. How are enzymes typically named? After the reaction they inhibit. Second, after their substrate or the reaction they catalyzed. Third, randomly assigned letters and numbers. And fourth, after their discovery. So we have seen it so far that they are named after their substrate or their reaction they catalyze. For example, if they act upon maltose, we name it as maltase. Okay. Similarly, if it acts upon the cellulose, we name it as cellulase. Therefore, the correct option would be option second. So in some cases, it is also named up after their reaction, like hydrolase, if it is doing a hydrolysis reaction. Fourth, the concentration of hormones in blood is usually in, so four options are there here. So first one is milligrams per liter, grams per ml, and then we have nanograms per milliliter, and lastly we have kilograms per milliliter. Okay, so the correct answer would be nanogram per milliliter, since it is present in a very small amount. So we cannot use this milligram or gram or kilogram for calculating the amount of hormones in blood. We use this nanogram. Next one, which of the following correctly describes the structural difference between a nucleoside and nucleotide? So we have talked a lot about this. A nucleotide contains a sugar and a base, while a nucleoside contains a sugar, a base, and a phosphate group. Okay. Second, a nucleoside contains a sugar and a base, while a nucleotide contains a sugar, base, and the phosphate group. Third, a nucleotide lacks a phosphate group, while a nucleoside includes one. Last one, both nucleosides and nucleotides contain phosphate group, but nucleotides have an additional sugar moiety. So we have discussed it. A nucleoside is that which contains only a sugar and a base, and your nucleotide contains an additional phosphate group apart from this sugar and base. Therefore, the correct option would be option second. Next one. In the secondary structure of DNA, adenine pairs with the thymine. Now, what is the number of hydrogen bond present between adenine and thymine? So, between your cytosine and your guanine, we have 3 hydrogen bond, and between adenine and thymine, we have 2 hydrogen bond. Therefore, the correct answer would be second. Of course, 1 and 2 hydrogen bonds are ultimately wrong, and covalent bonds are not present between the 2 strands. After that, which of the following statements about RNA is incorrect? Remember, you have to pick the incorrect statement. RNA is always single stranded and cannot form double helix. So this is obviously correct. DNA is double helix and RNA is single helix. Second one, RNA contains ribose sugar instead of deoxyribose sugar. Okay, so this is completely right. Why? Because we are knowing it from its name as well. Its name is ribonucleic acid. Got it? And DNA is deoxyribonucleic acid. So deoxyribo means deoxyribose. So it's quite easy to get that. Next one, RNA includes uracil instead of thymine. So this is also correct. Your DNA contains ATCG, means adenine, thymine, cytosine, and guanine. In case of your RNA, your uracil is present instead of thymine. Lastly, RNA can fold back on itself to form a secondary structure. So this is something which is not seen in case of RNA. Next, the primary biological function of nucleic acid includes: first, energy storage of cellular processes; B, catalyzing metabolic reaction; C, self-replication and protein synthesis; and D, structural support for cell membrane. So no, we know it very well that nucleic acids are responsible for storing the genetic information which with they can self-replicate as well as synthesize protein for our human body. Got it? So correct option would be option C. Next one, you have to tell that which statement about DNA is incorrect. Remember, you have to select the incorrect statement. First one, DNA is responsible for maintaining genetic identity across generations. So this is the very basic definition of DNA. Then second, it can self-replicate it. Yes, and that is the reason they can store the genetic information by self-replication from parent to daughter. Third, proteins are directly synthesized by the DNA. So this is not correct. RNA is the one which is responsible for the synthesis of protein and not the DNA. Last, DNA carries the genetic information for protein synthesis. So this is also correct. So DNA carries the information, whereas RNA decodes this information and they synthesize the protein. Got it? So DNA is not the one responsible for synthesizing protein. Last one, which molecule acts as the intermediary between DNA and the protein synthesis? First one, ribosomal RNA, or also known as rRNA. Second one, transfer RNA, or also known as tRNA. And the last one, the messenger RNA or the mRNA. And the last one we have it as deoxyribonucleic acid or DNA. So what will be the correct answer for this question? So we know it very well that these three which are given in the above part are messenger, transfer and ribosomal. So these three are types of RNA. Okay. So of course this DNA will not be the answer, right? The thing which act as the intermediary part is your messenger RNA. It carries the message from the DNA onto the RNA. Means the decoded message of your DNA. It carries that message so that your RNA can synthesize the protein. So the correct answer would be option C. So this was all for today's class. So I would suggest you to please attempt this DPP with by yourself also. It will help you to boost your preparations. Thank you for joining the sessions. I hope you enjoyed it.
Hello everyone, good evening. Okay, so in today's lecture, we will cover three-dimensional geometry. Okay, so according to last 5 year, the chapter three-dimensional geometry, the chapter weightage is 5.1% for JEE examination. And if we do year-wise analysis, so in their 2024, total 2 questions came from this topic based on distance from point to line. That is shortest distance between two lines. Okay. In their 2023, again 2 questions based on shortest distance. Okay. And in their 2020, 3 questions came from the topic equation of a straight line in space and shortest distance between two lines and distance from point to line. Okay. In that 2021, 1 question from shortest distance, and in their 2020, 1 question of easy level based on image of a point in a line. Okay. So, in today's lecture, first we will discuss the coordinate of a point in space. Okay. Then we will talk about the section formula and internal division for section formula, external division, some important result, and then some important deduction. After that, we will learn about how to write an equation of a line in space, okay, in 3D space, okay, and angle between two intersecting lines. Then I will tell you about skew lines, what are they, and then shortest distance between two skew lines, distance or shortest distance between two parallel lines, okay, coplanar lines, condition for coplanarity of two non-parallel lines, and then what is plane, and then we will see how to write equation of plane in different forms, okay, and then some result based on plane, and how to find out the angle between a line and a plane, and some points, important points related to line and plane. Okay, so let's start. So first thing, what is 3D space? Okay, so see this figure, this is a figure of 3D space. In comparing to 2D space, there are total 3D space, so 3 axes, x-axis, y-axis and z-axis. In 2D space, there are only 2 axes, x and y-axis, which are perpendicular, but there are total 3 mutually perpendicular axes, that is x-axis, y-axis and z-axis. Okay, and these all 3 axes make your 3D space. Okay, and this all three axes divide the space into total 8 parts. Okay, so the 3 mutually perpendicular lines, that mean your x-axis, y-axis and z-axis, divide the space into 8 parts and are called coordinate axes. So x-axis, y-axis and z-axis are called coordinate axes. So imagine a space which has 3 dimensions like length, width and height. So if there is a cube, then cube is a 3D space because it has total 3 dimensions. It has length, height and width. Total 3 axes, and each axis represent 3 dimensions. Okay. And the coordinate of point, just like if you want to find the coordinate of, as in figure, coordinate of C, that is 0, 0, Z. So OC is the perpendicular distance of point C. Okay, and P is any point on space, and its coordinate is given by x comma y comma z. Here x represent the distance of point P along x-axis. Y represent distance of this point P along y-axis, and Z represent distance of point P along z-axis. So that's how we can write coordinate of a point on a 3D space by using its three distance of on three axes. Okay, and coordinate of a point on x, y, and z axis. So these coordinates are given by x comma 0 comma 0, and if the point lies on y axis, then the coordinate is given by 0 comma y comma 0, and if the point lies on z axis, then it is given by 0 comma 0 comma z. Okay, it means if a point lies on x axis, then the y coordinate and z coordinate is 0. In similar way, if the point lies on z axis, then x coordinate and y coordinate is 0. So that's how we can write a points on axis. Okay, and the coordinate of any point P in space is given by x comma y comma z. Okay, and if there are any 2 points on this 3D space, let A and B are 2 points in the 3D space, and the coordinate of A is x1, y1 and z1, and coordinate of B is x2, y2 and z2, then the distance AB is given by root under x2 minus x1 whole square plus y2 minus y1 whole square plus z2 minus z1 whole square. Okay, so this is your distance formula. Okay, next is section formula. Okay, so let's suppose now we have 2 points P and Q. Coordinate of P is x1, y1 and z1, and coordinate of Q is x2, y2, z2. So these 2 points lies on 3D space. Okay, and now there is another point that is M, x, y, z, and this point divide the line joining the point P and Q in the ratio m is to n. So the point M can divide into two way. One is either internally or externally. So if the point M divide the line joining in internally, okay, then the coordinate of M is given by the formula mxyz is equal to mx2 plus nx1 by m plus n. So x coordinate is mx2 plus nx1 by m plus n. Its y coordinate is given by my2 plus ny1 by m plus n, and its z coordinate is given by m into z2 plus nz1 by m plus n. Okay, so if in the 3D space any point divide the line joining of any other 2 point in the ratio m is to n, then that's how we can find the coordinate of that point M. Okay, and if the point M divide externally in the ratio m is to n, then the external point coordinate is given by x coordinate is given by mx2 minus nx1 by m minus n, y coordinate is given by my2 minus ny1 by m minus n, and z coordinate is given by mz2 minus nz1 by m minus n. Okay, clear? And if M lies in midpoint of the line joining P and Q, okay, then the coordinate of the midpoint is given by x1 plus x2 by 2, comma y1 plus y2 by 2, comma z1 plus z2 by 2. Okay, so some important result. So let's suppose we have a triangle ABC with vertex A, B and C. Okay, and it lies on a 3D space, and coordinate of A, that means vertex A is x1, y1, z1, and vertex B is x2, y2, z2, and vertex C is x3, y3, z3. Okay, then centroid of this triangle, okay, when we draw the median for each vertex and where they intersect all three median, that point is your centroid, and the coordinate of centroid as given in the figure, that coordinate is given by x1 plus x2 plus x3 by 3, comma y1 plus y2 plus y3 by 3, comma z1 plus z2 plus z3 by 3. Okay, and area of this triangle ABC is given by, it is given by this determinant, that is 1 by 2 into determinant of first row is your I, j and k unit vector, and second row is x2 minus x1, y2 minus y1, z2 minus z1, and third row is x3 minus x1, y3 minus y1, z3 minus z1. So if we have the coordinate of vertices of a triangle, then by using the determinant we can find the value of area of the triangle. Okay, and we know that if this area is 0, that means points A, B and C are collinear. Okay, next result is based on tetrahedron. So what is tetrahedron? Tetrahedron is a shape that is made by the 4 triangles. Okay, so 4 non-coplanar points A, B, C and D. Coordinate of A is x1, y1, z1, B is x2, y2, z2, C is x3, y3, z3, and coordinate of D is x4, y4, z4. They form a tetrahedron, and the vertices of tetrahedron are A, B, C and D. And edges are, first is edge is AB, then AC, AD, BC, BD and CD. It has as a 3D shape, so it has faces. First faces is ABC, one triangle, ABD, second triangle, ACD, third triangle, and BCD, fourth triangle. So by using this fourth triangle, we can say this is faces of a tetrahedron. Okay. So centroid of tetrahedron, as we can see in the figure, the red S is the centroid of tetrahedron. Okay, when we draw the median through a vertex D to the opposite face, that is that SABC is a centroid of a triangle ABC, and that line D to S is median to the vertex D to the opposite face ABC. And when we draw for all 4 vertices, where they intersect, that point is your centroid of tetrahedron, and this coordinate of this centroid is given by x1 plus x2 plus x3 plus x4 by 4, comma y1 plus y2 plus y3 plus y4 by 4, comma z1 plus z2 plus z3 plus z4 by 4. Okay, and volume of tetrahedron is given by, we use the determinant to find the volume, and that is 1 by 6 times the determinant, whose first row is x2 minus x1, y2 minus y1, z2 minus z1. Second row is x3 minus x1, y3 minus y1, z3 minus z1. And third row is x4 minus x1, y4 minus y1, and z4 minus z1. Okay, so this is all about tetrahedron. Now we'll talk about direction, cosine and ratio. Okay, so suppose a 3D space with x-axis, y-axis and z-axis, and let's suppose there is a point P on the space, and the coordinate of point P is x, y and z. That is, the distance of point P along x-axis is x, along y-axis is y, and along z-axis is z. And this line OP make an angle alpha, beta and gamma with positive direction of x, y and z axis respectively. It means this line make alpha angle with positive direction of x axis, make beta angle with positive direction of y axis, and make gamma angle with positive direction of z axis. Okay, so when we take the cos of this angle, then we get direction cosine. Okay, so cos alpha, cos beta and cos gamma are called direction cosines, and they are denoted by L, M and N. Okay, so L is equal to cos alpha, M is equal to cos beta and N is equal to cos gamma. These are direction cosines. And what about direction ratio? So if number A, B, C, any number, and these number are proportional to direction cosines, that is proportional to L, M and N, then this number A, B and C are called direction ratio. Okay, it means as these are proportional, so if A, B, C are the direction ratio of a vector, then and L, M, N are the direction cosine. So as they are proportional, so L by A is equal to M by B is equal to N by C. Okay. Now, a line in space can be expanded in two directions. Okay. So it has two sets of direction cosine. And to find a unique direction cosine, we must take the given line as directed line. It means we have to fix the direction of that line, because in a space a line can be extended in two directions, and according to the direction we can find the two direction cosine, and to get a unique direction cosine we have to take directed line. That means a line with direction. Okay, so let L is a directed line and which makes an angle alpha, beta and gamma with positive direction of x, y and z axis and respectively. And if we reverse the line, we reverse the direction of line L, okay, then the angle alpha, beta and gamma, that is your direction angle, are replaced by the supplements angle. So now the angles are pi minus alpha, pi minus beta and pi minus gamma. So we get another direction cosine according to this angle pi minus alpha, pi minus beta and pi minus gamma. Okay, so we have two direction cosine for each line in space. But if we have a directed line, it means if we have a line with direction, then we have unique direction cosine. Okay, so now let's suppose we have a line which does not passes through the origin. If the line does not passes through the origin, then how we can find the direction cosine of this line? Okay, so we use a concept that direction cosine of two parallel lines have same set of direction cosine. Okay, so if we take a parallel line that is passes through the origin and we find the direction cosine of that line, then the direction cosine of this new line that is passes through the origin and parallel to the given line is equals. Okay, so let's suppose we have 2 points on a space P and Q. P is x1, y1, z1 and Q is x2, y2, z2. And we join this 2 point, we get a line. And the direction cosine, direction ratio of this line is given by, so if we have the 2 points on a line, then direction ratio is given by x2 minus x1, y2 minus y1 and z2 minus z1. And its direction cosine is given by x2 minus x1 by pq, magnitude of pq, that is distance pq, comma y2 minus y1 mod pq, comma z2 minus z1 by mod pq. And this is your distance. Mod pq is distance of point P to Q, and that we can get by the distance formula. Okay. Now let P be any point, P x y z be any point in space, and OP is R, that is distance of point O. Position vector of point P is R. Okay. Then we want to write this position vector OPR in terms of direction cosine. Okay, so first thing we can write x as L into mod R, y is equal to m into mod R and z is equal to n into mod R. Okay, and here L into mod R and m into mod R and n into mod R are projection of R on OX, OY, on OZ respectively. Okay, so we can write the point OP, that is a position vector of point P in space. So R can be written as R vector is magnitude of R into Li plus Mj plus Nk, where this Li, Mj and Nk are the unit vector of vector R. Okay, so that's how we can identify the point P on a space by using direction cosine. Okay, and if we have a vector R, that is mean the position of a point in a vector form, that is R is equal to AI plus BJ plus CK, then ABC are the direction ratio of vector. So if we have a point in the vector form, in a component form, then we can easily find the direction ratio that are the A, B and C, that is scalar component are the direction ratio. And the direction cosine is given by the formula L is given by A by magnitude of R, M is equal to B by magnitude of R, and N is equal to C by magnitude of R. Okay. And we have one property based on direction cosine is that if L, M and N are the direction cosine, and if we take the square of all three and take a sum, then it is equal to unity. That means L square plus M square plus N square is equal to 1. Okay. One important thing, direction cosine are unique. Okay. If we have a directed line, then direction cosine are unique, but direction ratio are not unique. Okay. And it can be infinite. Okay. So if you have a direction cosine like ABC and you take any scalar multiple of the direction cosine like KA, KB, KC, where Kn is not equal to 0, then all are the direction ratio. So direction ratio are not unique and it can be infinite. And if we have the direction cosine, direction ratio of line, like if ABC are the direction ratio of line, and let LMN are direction cosine of line, then we can find the direction cosine by using the direction ratio, and that is given by L is equal to plus minus A by root under A square plus B square plus C square. M is equal to plus minus B by root under A square plus B square plus C square. And N is equal to plus minus C by root under A square plus B square plus C square. Okay. Next thing. Now let's suppose if a line is equally inclined to coordinate axis, that means the line makes the angle with positive direction of x-axis, y-axis and z-axis all are equal. Okay, then direction cosines are given by plus minus 1 by root 3, comma plus minus 1 by root 3, comma plus minus 1 by root 3. That means the value of L, M, N are plus minus 1 by root 3. And we can easily find this coordinate by using a simple relation, that is L square plus M square plus N square is equal to 1. And as if we take the same angle, then L is equal to cos alpha, M is equal to cos alpha, N is equal to cos alpha. As the line is equally inclined to coordinate axis, so we get 3 cos square alpha is equal to 1, and from that we get the value of L is plus minus 1 by root 3 and plus minus 1 by root 3 M, and N is plus minus 1 by root 3. Okay. And if L, M, N are the direction cosine of a line, then maximum value of the product of the direction cosine, that means the product L into M into N. So maximum value of this product is 1 by 3 root 3. Okay. Okay. So now we'll talk about equation of a line in space. Okay. So how we can write a equation of a line in space? Okay. So to write equation of line in space, we have a point on a line, and we also need direction of that line. Then we can write equation of line. And if we also have 2 points on the line, then we can also write the equation of line. So first case, if equation of line passing through a point, that means if we have a point on a line, that is point A, and the position vector of this point A is A. Okay. And parallel to vector B, that means this line is parallel to vector B. So as this is parallel to a vector, so the direction of this line is same as that parallel vector. So direction of this line is B vector. Then the vector form of equation of line is R is equal to A plus lambda B, where R is any arbitrary position vector on a line. Okay. And in Cartesian form, if we have the coordinate of A, that is x1, y1, z1, and A lies on the line, and the direction ratio of the line is ABC, then equation of line in Cartesian form is x minus x1 by A is equal to y minus y1 by B is equal to z minus z1 by C. Okay. So equation of x-axis. So we have to find equation of x-axis. So as this line passes through origin, so we have a point on the line that is 0 comma 0 comma 0. So the value of x1 y1 z1 is 0 0 and 0. Okay, and as this line is parallel to your unit vector I, and direction ratio of unit vector is 1 comma 0 comma 0, so we have the value of direction ratio. The value of A is 1, value of B is 0, and value of C is 0. So equation of line, by using the Cartesian form of equation of line, we get x minus 0 by 1 is equal to y minus 0 by 0 is equal to z minus 0 by 0, or we can also write the equation of x-axis as y is equal to 0 and z equal to 0. Okay, and what about equation of y-axis? In a similar way, this line is parallel to j, unit vector j. So direction ratio is 0 1 0. So we get x minus 0 by 0 is equal to y minus 0 by 1 is equal to z minus 0 by 0, or x equal to 0 and z equal to 0. And equation of z-axis is x minus 0 by 0 is equal to y minus 0 by 0 is equal to z minus 0 by 1, or we can write the equation of z-axis as in this axis, if there is any point lies on z-axis, then its coordinate is 0 comma 0 comma z. That means the value of x coordinate and y coordinate on a z-axis is always 0. So we can write the equation of z-axis as x equal to 0 and y is equal to 0. In a similar way, as in 2D space, we write the equation of x-axis as y is equal to 0 and equation of y-axis as x equal to 0. Okay. Now let's suppose we have a line on a space, and also we have a 2 points on the line, and the first point is A x1 y1 z1 and second point is B x2 y2 z2. So the line is passing through these 2 points. Then equation of line is given by x minus x1 by x2 minus x1 is equal to y minus y1 by y2 minus y1 is equal to z minus z1 by z2 minus z1. Okay, we know that if we have the 2 point and we join those 2 point to get a line, then direction ratio of the line is given by x2 minus x1, y2 minus y1 and z2 minus z1. So this form is nothing but the form x minus x1 by A is equal to y minus 1 by B is equal to z minus z1 by C. We just substitute the value of A, B and C. Okay, and in the vector form, if we have the 2 points on the line, then vector form is R is equal to A plus lambda time B minus A. Okay, and in parametric equation of line, that is passes through a point A1, A2, A3, and the direction cosine of this line is LMN, then the parametric form of equation of line is x equal to A1 plus LR, y is equal to A2 plus MR, and z equal to A3 plus NR. Okay, so now we'll discuss how to find the angle between two given lines. Okay, so for finding the angle, let's suppose direction ratio of two lines are A1 B1 C1 and A2 B2 C2. Okay, so if we have the direction ratio of the both line and let the theta be the angle between these two lines, then it is given by cos theta is equal to A1 into A2 plus B1 into B2 plus C1 into C2 by root under A1 square plus B1 square plus C1 square into A2 square plus B2 square plus C2 square. So if we have the direction ratio of both line, then we can find the angle between this two line by using this formula. Okay, so condition for parallel lines, that means these two lines are parallel if A1 by A2 is equal to B1 by B2 is equal to C1 by C2. So if we have two line and direction ratio of these two lines are A1 B1 C1 and A2 B2 C2 respectively, then if they satisfy this ratios condition, that is A1 by A2 is equal to B1 by B2 is equal to C1 by C2, then both lines are parallel. And what about perpendicular? So condition of perpendicular is A1 A2 plus B1 B2 plus C1 C2 is equal to 0. Okay, but if we have the direction cosine of the lines, then let's suppose we have two lines and direction cosine of first line is L1, M1 and N1, and for second line it is L2, M2 and N2. Then angle between these two lines is given by theta is equal to cos inverse L1 L2 plus M1 M2 plus N1 N2, or it in terms of sine, it is given by theta is equal to sine inverse root under m1 n2 minus m2 n1 whole square plus n1 l2 minus l1 n2 whole square plus l1 m2 minus l2 m2 whole square. Okay. And condition of parallel lines in terms of direction cosine, and this is L1 by L2 is equal to M1 by M2 is equal to N1 by N2. And for condition for perpendicular line in terms of direction cosine, this is L1 L2 plus M1 M2 plus N1 N2 is equal to 0. Okay. So some result based on angle between two lines. So the angle between any two diagonal of cube is given by cos inverse 1 by 3. Second result, angle between a diagonal of a cube and a face is cos inverse root under 2 by 3. Third result, the angle between the diagonal of a cube and edge of a cube is cos inverse 1 by root 3. And if a straight line make angle alpha, beta, gamma and delta with diagonal of cube, then cos square alpha plus cos square beta plus cos square gamma plus cos square delta is equal to 4 by 3. These are the four results related to angle between two intersecting lines. Okay. An angle between lines and also plane. Okay. Now, now we'll talk about skew lines. Okay. So in case of 2D, there's two types of line. First is intersecting and another is parallel lines. But in case of 3D space, there are three types of line. Either it is intersecting, it can be parallel, or it can be skew lines. Okay. So two straight line in space which are neither parallel nor intersecting are called skew lines. Okay. So skew lines are the lines which are not coplanar lines. It means they do not lie in the same plane. Okay. As in this figure, the line P, there's two plane P and Q and two line A and B. So these two lines does not lie in a same plane. Okay. And these lines are not even parallel line, as the direction of both line is not same. So these lines are not parallel, and as these lines lies on two different plane, so they cannot intersect. Okay. So these lines are skew lines. Okay. So how we can find the shortest distance between two skew lines? Okay. So let's suppose we have two skew lines L1 and L2 and we have to find the shortest distance. So there are only one line that is perpendicular to both line. Okay. That means we can draw a line that is perpendicular to line L1 and line L2. And the distance of this perpendicular is your shortest distance between these two skew lines L1 and L2. Okay. So the shortest distance between two lines L1 and L2 is distance PQ between the point P and Q, where the line of shortest distance intersect the two given line. Okay. So to find the shortest distance between two skew line, let L1 and L2, we draw a perpendicular that is perpendicular to both line L1 and L2. So let's suppose that that perpendicular line is PQ. So P is the point which lies on line L1 and Q is the point which lies on line L2, and PQ is the shortest distance between these two skew line. Okay. So if we have a equation of line in vector form of two skew lines, so R is equal to A plus lambda B is your first line and R is equal to C plus mu D is your second line. So these are two skew lines. So the shortest distance, that is SD, of these two skew line is given by the formula is mod C minus A dot B cross D by magnitude of B cross D. Okay. And if the equation of line given in Cartesian form, so the shortest distance between the lines x minus x1 by a1 is equal to y minus y1 by b1 is equal to z minus z1 by c1, and x minus x2 by a2 is equal to y minus y2 by b2 is equal to z minus z2 by c2 is given by this. Shortest distance SD is equal to mod of determinant x2 minus x1, y2 minus y1, z2 minus z1, a1 b1 c1, a2 b2 c2 by, in denominator, root under b1 c2 minus b2 c1 whole square plus c1 a2 minus c2 a1 whole square plus a1 b2 minus a2 b1 whole square. Okay, so we have to remember this formula to find shortest distance between two skew line. If it is given in vector form, then the formula is mod of c minus a dot b cross d by magnitude of b cross d, and if it is given in Cartesian form, then we can calculate by using this formula, by using that is a determinant, by using determinant. The numerator part is determinant, and in the denominator part, the root under b1 c2 minus b2 c1 whole square plus c1 a2 minus c2 n whole square plus a1 b2 minus a2 b1 whole square. Okay, so the two lines are a plus lambda b and r is equal to c plus mu d are intersecting. That means these lines are intersecting. Then the shortest distance is 0 because lines are intersecting. So in this case, this formula, the shortest distance is equal to 0. So mod c minus a dot b cross d by magnitude of b cross d is equal to 0. And from here we get c minus a dot b cross d is equal to 0. This is the condition for intersect two line. Okay. And in case of Cartesian form, if this determinant is 0, then two line are intersecting. That is x2 minus determinant of x2 minus x1, y2 minus y1, z2 minus z1, a1 b1 c1, and third row a2 b2 c2 is 0. Okay. Now, shortest distance between two parallel lines. Okay. So if we have two parallel lines, then how we can find the shortest distance? So again, the same approach, we draw a perpendicular through this both line, and then we find the distance of this perpendicular line. Okay, length of this perpendicular. Okay, so the shortest distance between parallel line will be the perpendicular distance. So if we have two parallel lines in vector form, that is r is equal to a plus lambda b and r is equal to c plus mu b. B is the direction vector, and as these lines are parallel lines, so direction is same. So we get equation as r is equal to a plus lambda b and r is equal to c plus mu b. Then distance between them is given by this formula, d is equal to mod c minus a cross b by magnitude of b. Okay. Next is coplanar lines. So coplanar, that means lines lie on the same plane. Then lines are called coplanar lines. Okay. And if lines lies on same plane, then these lines can never be skew lines, because to be a skew line must be lie on two different plane. So if a line lies on a same plane, then either it is intersecting or parallel. Okay. So condition for coplanarity of two non-parallel lines. So if we have two lines and these lines are non-parallel, so direction must be different. So the vector form is r is equal to a plus lambda b, first line, and second line is r is equal to c plus mu d, are coplanar or intersecting if condition is c minus a dot b cross d is equal to 0, and it implies that scalar triple product of ABD is equal to scalar triple product of CBD. So this is the condition for two lines are coplanar. Okay. And in Cartesian form, so if the equation of lines are x minus x1 by L1 is equal to y minus y1 by m1 is equal to z minus z1 by n1, and x minus x2 by L2 is equal to y minus y2 by M2 and z minus z2 by N2 are coplanar if this determinant is equal to 0. That means determinant of x2 minus x1, y2 minus y1, z2 minus z1, L1 m1 n1, L2 m2 n2 is 0. Okay. Next is plane. So how we can define a plane? So plane is a surface. If we take any 2 points in a plane and if we join this 2 point, then we get a line, and if the all points on that line must lie on a plane, totally lies on a plane, then this surface is called plane. Okay, so plane is a surface such that if we take any 2 point on that surface and we join this 2 point, we get a line. Okay, and if this line is completely lies on the surface, then that surface is called plane. Okay, so now we'll discuss different form of equation of plane. So first, general equation. So general equation of plane is ax plus by plus cz plus d is equal to 0, provided that a square plus b square plus c square not equal to 0. And here a, b, c are the direction ratio of the normal to the plane. Okay, so to find the general equation of plane, we have a normal to the plane and we also have the direction ratios of the plane, that is a, b, c. Then the general equation is ax plus by plus cz plus d is equal to 0, where d is a constant. Okay, and let's suppose this plane is passes through origin, then equation become ax plus by plus cz is equal to 0, constant become 0, and plane parallel to coordinate planes. Okay, so if the plane is parallel to y or z plane, that means yz plane, then equation of plane is x is equal to k. And if the plane is parallel to zx plane, zox plane, then equation of plane is y is equal to k. And if the plane is parallel to xy plane, then equation of plane is z is equal to k. Okay, and now planes parallel to coordinate axis. So if your plane is parallel to x-axis, then equation become by plus cz plus d is equal to 0. If it's parallel to y-axis, then it become ax plus cz plus d is equal to 0. If it is parallel to z-axis, then become ax plus by plus d is equal to 0. Okay, and if the intercept of the plane on the coordinate axis are a, b, and c, then equation of plane is given by x by a plus y by b plus z by c is equal to 1. Here a, b, c are the intercept of the plane with the coordinate axis. Okay, and it meet the coordinate axis, that means the plane meet the coordinate axis at a. On x-axis it meet at a comma 0 comma 0. It intersect, that means it meet on y-axis at a point b, that is 0 comma b comma 0, and on z-axis is c, that is 0 comma 0 comma c, intercept point. Okay, and if L, M, N are the direction cosine of normal to the plane. Okay, so now we have direction cosine of normal, that is to the plane, and P is the distance of origin from the plane. Okay, then equation of plane is given by Lx plus My plus Nz is equal to P. So in this equation, L, M, N are the direction cosine of normal to the plane, and P is the distance of this plane from origin. Okay, and if we draw a perpendicular from the origin, then foot of perpendicular is given by LP, MP, NP. Okay, now let ON is normal from origin to the plane. We draw a normal to the plane, and that is ON, and N cap is unit vector along ON. Then ON is the distance from origin to the plane is P. So ON is, as the unit vector is N cap, so ON is P times N cap. Okay, and equation of plane, in this case equation of plane is given by R dot N cap is equal to P. Okay, where R is xi plus yj plus zk. Okay, so these are the different form of equation of plane. Next is, if plane through a point, we draw a plane through a point x1 y1 z1, and A, B, C are the direction ratio of normal to the plane. Then equation of plane is A into x minus x1 plus B into y minus y1 plus C into z minus z1 is equal to 0. And if we draw a plane through three non-collinear points x1, y1, z1, x2, y2, z2 and x3, y3, z3, then we get that is determinant of x1 minus x1, y minus y1, z minus z1. Second row x2 minus x1, y2 minus y1, z2 minus z1. Third row x3 minus x1, y3 minus y1, z3 minus z1 is equal to 0. So this is the equation of plane when we draw a plane through three non-collinear points. Okay. 6th point, equation of plane passing through a point A and with position vector A and parallel to a given vector B and C. Okay, so now we have to find equation of plane that is passing through a particular point A, and the position vector of that point A is A, and this plane is parallel to any two given vector B and C. Then in this case, equation of plane is R minus A dot B cross C is equal to 0, or scalar triple product of R minus A, B, C is 0. Okay. In Cartesian form, we write this as by using determinant, that is determinant of x minus a1, y minus a2, z minus a3, second row b1, b2, b3, and third row c1, c2, c3 is 0. Okay. And if we draw a plane parallel to a given plane ax plus by plus cz plus d is equal to 0, then the equation of parallel plane is ax plus by plus cz plus k is equal to 0. Only term is different is constant term, that is k. Okay. And that value of k is determined by the given condition. Next, if we draw a plane that is passing through the line of intersection of plane ax plus by plus cz plus d is equal to 0 and a1x plus b1y plus c1z plus d1 is equal to 0, and we draw a plane through the intersection. So when we intersecting two plane, we get a line. Okay. And when then we draw a plane through this line of intersection. So the equation of that plane is given by x plus by plus cz plus d plus lambda time a1x plus b1y plus c1z plus d1 is equal to 0. Okay. And if r into n1 is equal to d1, if we have the equation of two plane in vector form, that is r into n1 is equal to d1 and r into n2 is equal to d2, two plane, there n1 and n2 are the normal to the plane. Okay. Then their line of intersection is perpendicular to both n1 and n2. Okay. So when we intersecting these two plane, we get a line, and that is line of intersection. So this line of intersection is perpendicular to both n1 and n2. That means, so perpendicular to both n1 and n2. So to find this line, we cross the product. So n1 cross n2 is the line which is perpendicular to n1 and n2 both. So that means the line of intersection is parallel to n1 cross n2. Okay. So some important result on plane. So if ax plus by plus cz plus d1 is equal to 0 and a1x plus b1y plus c1z plus d2 is equal to 0 are equation of two plane. Okay. So when they intersect, that means when we equate both equal, that is ax plus bx plus cz plus d1 is equal to a1x plus b1y plus c1z plus d2, then we get a straight line. That means when we intersect two plane, we get a line. As in 2D, when we intersect two lines, we get a point. But when we intersect two plane, we get a line, a straight line. A plane, let's suppose we have a plane ax plus by plus cz plus d is equal to 0, and this plane intersecting a line joining two point a x1 y1 z1 and b x2 y2 z2. So this plane divide this line in the ratio minus ax1 plus b1y plus cz1 plus d by ax2 plus by2 plus cz2 plus d. Okay. And if this ratio is positive, then this point a and b are on opposite side of the plane, and if it is negative, then the point a and b are on same side of the plane. Okay. And if you want to find the angle between two planes, so we have two planes, and let's suppose there is a angle between these two planes is theta, then theta is given by cos inverse mod a1 a2 plus b1 b2 plus c1 c2 by root under a1 square plus b1 square plus c1 square into root under a2 square plus b2 square plus c2 square. Okay. And two planes are parallel if their normals are parallel, and two planes are perpendicular if their normals are perpendicular. Okay. Next, if r into r dot n1 is d1, that means if we have two plane r dot n1 is equal to d1 and r dot n2 is equal to d2, then they are parallel if their normals are parallel. That means if n1 and n2 are parallel, that means n1 is equal to lambda time n2. Or in Cartesian form, if a1 by a2 is equal to b1 by b2 is equal to c1 by c2. And if two planes are perpendicular, if their normal are perpendicular, that means n1 and n2 are perpendicular, and to n1 and n2 are perpendicular, the dot product is 0, that is n1 dot n2 is 0, and in Cartesian form, a1 a2 plus b1 b2 plus c1 c2 is equal to 0. Okay, by using the direction ratio of the normals. Now, to find the distance of this point x1 y1 z1 from the plane ax plus by plus cz plus d is equal to 0, this is given by mod ax1 plus by1 plus cz1 plus d by root under a square plus b square plus c square. And distance of origin from the plane is given by mod d by root under a square plus b square plus c square. And to find the distance between two parallel plane, so that is given by mod d1 minus d2 by root under a square plus b square plus c square. Okay. And the angle between the line and a plane. So if we have a equation of line in Cartesian form, x1 minus x1 by a is equal to y minus y by b is equal to z minus z1 by c. And we also have a plane, that is a1x plus b1y plus c1z plus d is equal to 0. And the angle between them, let's consider it is theta. Then 90 degree minus theta is angle between normal and a line. Okay. Then that is given by cos 90 minus theta is equal to mod a1 plus b into b1 plus c into c1 by root under a square plus b square plus c square into root under a1 square plus b1 square plus c1 square. Okay. And if a by a1 is equal to b by b1 is equal to c by c1, then line is perpendicular to the plane. And if a into a dot a1 plus b dot b1 plus c dot c1 is equal to 0, then line is parallel to the plane. Okay. And if a dot a1 plus b dot b1 plus c dot c1 is equal to 0 and a1x1 plus b1 y1 plus c1 z1 plus d is equal to 0, then line lies in the plane. So this is the result based on a line and a plane. Okay. So just wait for a moment. Now we'll practice some question. Okay. So first question is, if the orthocenter and centroid of triangle are minus 3, 5, 1 and 3, 3, minus 1 respectively, then its circumcenter is. Options are option A 6, 2, minus 2, option B 1, 2, 0, option C 6, 2, 2 and option D 6, minus 2, 2. Okay, so take your time and try this question. In what ratio centroid divide the line joining orthocenter and circumcenter? If you know the ratio, then apply the section formula to find the circumcenter. Okay. Okay, let's solve. Correct answer is option A, that is 6 comma 2 comma minus 2. The formula that we use, we know that the centroid G divide the line joining the orthocenter H and circumcenter O in the ratio 2 is to 1. Okay, so when we apply the section formula, we get G is equal to 2 into O plus H by 3. Okay, so now we have the coordinate of centroid and coordinate of orthocenter, and we have to find the coordinate of circumcenter. That means we have to find the value of O. Okay, so let's solve. Orthocenter is H, that is minus 3 comma 5 comma 1, and centroid, that is Z, that is 3 comma 3 comma minus 1. Okay, so we have a relation, that is Z is equal to 2 into O plus H by 3. So from here we get 3Z is equal to 2O plus H. So substitute the coordinate of G and H. So we get 3 into 3 comma 3 minus 1 is equal to 2 into O plus H, that is minus 3 comma 5 comma 1. Okay, so from here we get the coordinate of O as 9 plus 3 by 2 comma minus 5 plus 9 by 2 comma minus 3 minus 1 by 2. That is coordinate of circumcenter is 6 comma 2 comma minus 2. So important thing is the centroid G divide the line joining the orthocenter and circumcenter in the ratio 2 is to 1. Okay. Second question. A line makes the same angle theta with each of x and z axis. Okay. And if the angle beta which it makes with y axis, so there is a line in space, and that line makes angle theta with x and z axis and make angle beta with y axis. Okay, is such that sine square beta is equal to 3 sine square theta. So there is a relation on beta and theta, that is sine square beta is equal to 3 sine square theta. Then what is the value of cos square theta? Options are option A 2 by 3, option B 1 by 5, option C 3 by 5, and option D 2 by 5. So try this question. Just recall what is the relation between L, M and N, where L, M, N are the direction cosine of a line if it make an angle alpha, beta and gamma with positive direction of x, y and z axis. Okay, so try this question. What is the relation between L, M and N? Anyone? That relation is L square plus M square plus N square is equal to 1. Okay, so use this relation. Okay, let's solve. So correct answer is C, that is 3 by 5. Okay. So we know that what is direction cosine? So if there is a line in 3D space and that line makes an angle alpha, beta and gamma with the positive direction of x, y and z axis, then direction cosine are L is equal to cos alpha, M is equal to cos beta, N is equal to cos gamma. Okay. And the sum of square of direction cosine, that is always equal to 1. That is L square plus M square plus N square is equal to 1. Okay. So let's solve. It is given that line makes angle theta with x and z axis, okay, and beta with y axis. So L is cos theta, M is cos beta, and N is cos, again cos theta. Okay. So we know that L square plus M square plus N square is equal to 1. So substitute this value. So we get cos square theta plus cos square beta plus cos square theta is equal to 1. Okay. So from here we get 2 cos square theta is equal to 1 minus cos square beta. And we know that 1 minus cos square beta is sin square beta. So we get our equation 1, that is 2 cos square theta is equal to sin square beta. Okay. Now it is also given that sin square beta is equal to 3 sin square theta. So from equation 1 and 2, we get 3 sin square theta is equal to 2 cos square theta. And we can write the sin square theta as 1 minus cos square theta. So now my equation is all are in cos theta. So we get 3 is equal to 5 cos square theta. We have to find the value of cos square theta. So from here cos square theta is equal to 3 by 5. Clear? Simple. Next, question number 3. A line makes an angle theta with x and y axis both. A possible value of theta is in, so there is a line in space and it make an angle theta with x and y axis. Okay. And we have to find the possible value of theta. Okay. Option are option A, close interval 0 to pi by 4. Option B, close interval 0 to pi by 2. Option C, close interval pi by 4 to pi by 2. Option D, close interval pi by 3 to pi by 6. Okay. So try this question. We use the same relation, L square plus M square plus N square is equal to 1 in this question. Okay. So try this. Okay, option C. So correct answer is option C. So we know that if there is a line in 3D space and it makes an angle alpha, beta and gamma with positive direction of x, y and z axis, then direction quotient is given by L is equal to cos alpha, M is equal to cos beta, N is equal to cos gamma. Okay, and there is a relation, that is L square plus M square plus N square is equal to 1. Okay, so we just use this equation to find the possible value of theta. Now it is given that this line make an angle theta with x and y axis. So L is equal to cos theta and M is equal to cos theta. Okay, and we know that L square plus M square plus N square is equal to 1. So let's suppose this line make an angle gamma with z axis. So we get cos square alpha, cos square theta plus cos square gamma is equal to 1. Okay, and from here we get cos square gamma is equal to 1 minus cos square theta minus cos square theta, that is cos square gamma is equal to sin square theta minus cos square theta. We know that 1 minus cos square theta is sin square theta. So, and what is sin square theta minus cos square theta? As cos square theta minus sin square theta is cos 2 theta. So we get cos square gamma is equal to minus cos 2 theta. Now left hand side is cos square theta, a positive value, and in the right hand side minus cos 2 theta. So to become positive, cos 2 theta must be negative, that is cos 2 theta is less than equal to 0. Okay, and what is the value of angle for which the cos theta is negative? Okay, so when the theta lies from pi by 2 to pi, the cos is negative. Cos theta is negative. That means 2 theta is lies between pi by 2 to pi. Okay, so theta is lies from pi by 4 to pi by 2. So we get close interval pi by 4 to pi by 2. Okay, you can draw the graph of cos theta to figure out the this interval. Okay, clear? Next question. The projection of a vector on the 3 coordinate axes are 6, -3 and 2 respectively. The direction cosine of the vector are, okay, option is 6 -3 2, option B 6 by 5 comma -3 by 5 comma 2 by 5, option C 6 by 7 comma -3 by 7 comma 2 by 7, option D -6 by 7 comma -3 by 7 comma 2 by 7. Okay, so try this question and state which option will be correct. Okay, again option C. Let's check. Very good. Correct answer is option C, that is 6 by 7 minus 3 by 7 comma 2 by 7. So the vector PQ is represented by the difference in coordinate of P and Q. Okay, so if we have a coordinate of P and coordinate of Q, then we can write the vector PQ as x2 minus x1 comma y2 minus y1 comma z2 minus z1, and the distance PQ is given by root under x2 minus x1 whole square plus y2 minus y1 whole square plus z2 minus z1 whole square. Okay. And the direction cosine of a vector are the cosine of angle that make an angle that makes a line with x and y and z axis. And we can calculate the cosine by using this formula. If we know the coordinate of P and Q, then direction cosine of the line PQ is given by x2 minus x1 by length of PQ vector, comma y2 minus y1 by PQ length of PQ vector, comma z2 minus z1 by length of PQ vector. Okay, so now we have the projection of vector on the three coordinate, that is 6, comma minus 3, comma 2. Okay, so let the vector be x2 minus x1, comma y2 minus y1, comma z2 minus z1. So as the projection is given, so these values are equal. So x2 minus x1 is equal to 6, y2 minus y1 is equal to minus 3, and z2 minus z1 is equal to 2. Okay, so now we have the value of x2 minus x1, y2 minus y1, and z2 minus z1. So we can also calculate the length of PQ. So the length of PQ is root under x2 minus x1 whole square plus y2 minus y1 whole square plus z2 minus z1 whole square, that is root under 36 plus 9 plus 4, that is 7. So direction cosine by using this formula is 6 by 7, comma minus 3 by 7 and 2 by 7. So correct answer is option C. Okay. Next question. If the projection of the line segment on the x, y and z axis in 3-dimensional space are 2, 3 and 6 respectively, then the length of the line segment is. Okay. So try this question. Options are option A 12, option B 7, option C 9 and option D 6. So similar type of question as question number 4. We just need to find the length of line segment. Okay. Versa, which option is correct? Okay, so correct answer is option B, that is 7. So in 3D space, if there is a line and the line segment has a projection A, B and C on x, y and z axis, then we can relate this projection with direction cosine, that is LR is equal to A, MR is equal to B and NR is equal to C, where R is the position vector of line, any point on the line. Okay. Next, the segment length R is calculated by R is equal to root under A square plus B square plus C square. Okay, and we also have a relation on direction cosine, that is L square plus M square plus N square is equal to 1. Okay, so let's look at the solution. Now it is given that this line, the projection of line segment on x, y and z axis are 2, 3 and 6. Okay, so we also use the last theory as in last question to find the length of this line segment. Okay, but in terms of direction cosine, it is given that LR is equal to 2, MR is equal to 3 and NR is equal to 6. Okay, and now we have a relation, that is L square plus M square plus N square is equal to 1. So by using this equation, we get LR whole square plus MR whole square plus NR whole square. Put the value. So we get 2 square plus 3 square plus 6 square, and that is L square plus M square plus N square, R square is equal to 4 plus 9 plus 36. And from here, as the value of L square plus M square plus N square is equal to 1, so we get R square is equal to 49. So the R is 7. Okay, simple. Next question. A vector R is inclined at equal angle to OX, OY and OZ. Okay. That means the vector R makes an angle, same angle with x-axis, y-axis and z-axis. Okay. If the magnitude of R is 6 unit, then R is equal to. So we have to find the vector R. Option A, root 3 I plus j plus k. Option B. Just recall the formula, how we can write, a vector R using direction cosine. Okay, let's solve together. So correct answer is C, that is minus 2 root 3 I plus j plus k. Okay. So we know that as it is given that R vector inclined angle alpha to each axis with the same angle, equal angle. So direction cosine is L is equal to M is equal to N, that is cos alpha. And we know that L square plus M square plus N square is equal to 1. So we use this. So we get 3 cos square alpha is equal to 1. So now we have two cases according to alpha. So if alpha is acute, then L is equal to M is equal to N is 1 by root 3 in case of alpha is acute. Okay. And it is given that magnitude of R is 6. And we know that we can write a vector in terms of direction cosine, that is magnitude of R into Li plus Mj plus Nk. So now we have all this value. So substitute. So we get 6 into 1 by root 3 I plus 1 by root 3 j plus 1 by root 3 k, that is 2 root 3 I plus j plus k. That is your R. Okay. And in case 2, if your angle is obtuse angle, so in this case the direction cosine are negative. So LMN is minus 1 by root 3 and mod R is 6. So we get R is equal to magnitude of R Li plus Mj plus Nk. When we substitute the value, we get R is equal to minus 2 root 3 I plus j plus k, which match with option C. So correct answer is option C. That is your vector which make an angle, equal angle with OX, OY and OZ axis with magnitude 6 unit. Okay, clear? So that's all for today. Today we discussed about three-dimensional geometry and we practice some question. Okay. Any doubt? Okay then. Thank you.
Hello students, in today's lecture we are going to learn about atoms. And students, this is theory part. This is our disclaimer. So if we talk about the trend analysis of this chapter according to last 5 years, so we can say chapter contains overall 2.48%. Okay, if we talk about yearly question distribution, so we can say in 2024 question asked from energy of electron in hydrogen atom. Number of questions 1. And if we talk about their difficulty level, so we can say question was average. Similarly, in 2023 question asked from Bohr model of the hydrogen atom, the line spectra of the hydrogen atom. Number of questions 2. One question was average and one question was easy. In 2022 question asked from energy of electron in hydrogen atom. Number of questions 1 and their difficulty level average. In 2021 question asked from atomic spectra. Number of questions 1 and question was easy. In 2020 question asked from Bohr model of the hydrogen atom, the line spectra of the hydrogen atom. Okay, number of questions 1 and their difficulty level average. All right, so this is the uh, yearly question distribution. Clear? All right. So students, in today's lecture we will cover first atoms, then we will cover scattering of alpha particles, then we will discuss Rutherford's model of an atom, then Bohr's model, then some characteristics of an atom. Clear? Then we will discuss energy levels and hydrogen spectrum. All right, so these are the topics which we will cover in today's lecture. So let's start today's first topic, atoms. So suppose you have a metal such as iron. Okay, you cut the iron once, twice, thrice and cut it so much that you get a particle that cannot be cut further. Okay, so the smallest particle in which that element, for example we are talking about iron here, possesses all the properties of iron, is called an atom. Okay, as I took the example of iron here, an atom is the smallest particle of any element which possesses all the properties of that element. Okay, basically we can say atom is the smallest particle. And if we talk about the molecules, so basically a molecule is a group of atoms. Clear? What is molecules? A group of atoms. A molecule is formed from atoms which are connected to each other through chemical bonds. Okay, and this is the smallest unit of a chemical compound that can exist independently on its own. And if we talk about the nuclei, so we can say nuclei refers to plural of the nucleus of an atom, which contains a fixed number of nucleons. Okay, which contains fixed number of nucleons. Nucleons means uh, uh, formi- uh, uh, protons and neutrons. Okay, an atom made up of protons and neutrons called nucleons. This is a general term used for all known nucleides, whether stable or unstable of any chemical element. Okay, if we talk about an example, so we can say O16 and O17 are different nucleides. This means if we talk about nuclei or a nucleus, an atom is understood to be composed of several things such as protons, neutrons, and electrons. Alright, so the particles in the center of this atom, such as protons and neutrons, when they combine from a central region where mass is concentrated. Okay, clear? Now we will discuss scattering of alpha particles. Alpha particle scattering is an experiment that Rutherford performed in 1911. Okay, with the help of this experiment, he proposed the atomic model, specifically the planetary model of the atom. Rutherford introduced the concept that an atom contains a nucleus. Now the question arises as to what will be the number of alpha particles scattered at at any angle theta. So that is, if our alpha particles are scattered at a certain angle theta, then how many particles will be scattered? N of theta is directly proportional to z square divided by sine raised to the power 4 theta by 2 k square. Okay. Here k is the kinetic energy of alpha particle and z is the atomic number of the matter. So observe the number of particles at a given angle is proportional to z square. Yes or no? And we know z is the atomic number of the metal, meaning where alpha particles are being bombarded. Okay, and k is the kinetic energy of alpha particle and z is the atomic number. If we talk about the closest approach, or we can say distance of closest approach, so we can say uh, inside an atom there is a nucleus which is positively charged. Okay, uh, since an alpha particle is also positively charged, when an alpha particle approaches the nucleus, a repulsive force acts between the two. The initial kinetic energy that the alpha particle possesses gradually starts converting into potential energy because of repulsive force. It continues to approach the nucleus until all its kinetic energy is converted into potential energy. Okay, So the distance at which all its kinetic energy is converted into potential energy is called the distance of the closest approach. So at the distance of closest approach, we assume the distance between the nucleus and the alpha particle to be r naught. Now the alpha particle has a charge of 2e and the nucleus has a charge of JE. Okay. So at this moment the potential energy will be uh, 1 upon 4 pi epsilon naught JE into 2e divided by r naught. Okay. Here we know JE is the charge of the nucleus and 2e is the charge of alpha particle. This potential energy arose when all the initial kinetic energy of the alpha particle, which was 1 by 2 mv square, was converted into potential energy at the distance of closest approach. So we can say 1 by 2 mv square is equal to 1 upon 4 pi epsilon naught JE into 2e divided by r naught. Alright. Now after solving, the value of r naught is obtained as r naught is equal to JE square divided by mv square pi epsilon naught is equal to 4k JE square divided by mv square. Okay. Now we will discuss Rutherford's model of an atom. Clear? So students, Rutherford's model, Rutherford's atomic model based on the scattering of alpha particles. Okay, Rutherford postulated this model of the atom. What kind of model did he propose? Let's see. So first, we can say an atom is a sphere with a diameter of approximately 10 to the power minus 10 meters. Its entire positive charge and most of its mass are concentrated in the central part of the atom, which is called the nucleus. Okay. And if we compare the sizes of the atom and the nucleus, the comparison is such that if the atom were the size of a football field, then placing a small pin in the middle of that field would represent its nucleus. And if we talk about the order of diameter, so we can say the diameter of the nucleus uh, is of the order of 10 to the power minus 15 meter. Students, if we talk about the space around the nucleus, so we can say the space around the nucleus is largely empty and electrons revolve around the nucleus in the same way. Same way means as planets revolve around the sun. Okay. And if we compare the motion within an atom, to the motion of planets. It appears as if the sun is at the center of the planet's motion and the planets are revolving around it. Similarly, the nucleus is at the center of the atom and electrons revolve around it. Okay. Next point. Uh, the electrostatic attraction of the nucleus provide a centripetal force to the bring uh, provide centripetal force to the orbiting electron. Okay. Now, as it has been stated that electrons revolve around the nucleus within the atom. Okay. So, when they are revolving around it, who is providing the centripetal force for their revolution? It is stated here that because the nucleus is positively charged and the electron is negatively charged, so the positive charge attracts the negative charge with a Coulomb force, and that Coulombian force provides the centripetal force. Okay. And the total positive charge on the nucleus is equal to the total negative charge of the orbiting electrons. Okay. So if we talk about the atom, then we can say then atom is neutral. Number of positive charge is equal to number of negative charge. The atom is neutral. So Rutherford explained many things about the atom. However, his model still had some drawbacks. Okay. So first, stability. We can say stability of the atomic model. It means what? It means Rutherford's nuclear model failed to explain atomic stability. An electron revolving around the nucleus is in accelerated motion. Yes or no? And the classical electromagnetic theory states such a charge should emit energy continuously. Okay. So as the electron keeps losing energy, it would eventually spiral into the nucleus and collapse the atom. Alright. Since atoms are stable in reality, so Rutherford's model could not account for their stability, highlighting its major limitation. Second, nature of energy spectrum. It means what? It means Rutherford's model could not explain the nature of atomic spectra. We can say classical theory predicted that revolving electrons should emit continuous radiation, so producing a spectrum like a rainbow with smoothly varying wavelengths. However, experiments show uh, hydrogen emits discrete spectral lines at specific wavelengths. Thus, Rutherford's model failed to account for the discrete emission spectrum observed in reality. Okay, so these two are the uh, drawbacks of the Rutherford's model. Okay, and this is very important for the exam. All right. Now, next model, Bohr's model. So, students, attempted to resolve the drawbacks of Rutherford's model and proposed a new model of atom. Bohr added some postulates to Rutherford's model of the atom. So, first, uh, the electrons revolves around the nucleus only in certain permitted orbits. He stated that the electron revolves around the nucleus but not just anywhere. It will only revolve in certain permitted orbits, meaning it has some fixed orbits within which it can revolve. Okay. Now, how do we find these certain permitted orbits? So, Bohr stated that these certain permitted orbits are those where the electron's angular momentum is equal to an integral multiple of h upon 2 pi, where h is the Planck's constant. Clear? That is L is equal to mvn Rn is equal to nh upon 2 pi. He stated that if an electron is revolving in a certain orbit, which certain permitted orbit would that be? It would be an orbit where suppose the radius is r n and the velocity is vn and the electron's mass is m. Then if we calculate the angular momentum, it will be mvn Rn is equal to nh upon 2 pi. Okay. And the electrons do not radiate energy. The second point, the electrons do not radiate energy while revolving in the permitted orbits. That is, the permitted orbits are stationary, non-radiating orbits. Okay. Next, now he stated that energy is radiated only when an electron jumps from outer permitted orbit to an inner permitted orbit. Okay. So suppose there is a hydrogen nucleus which with one proton present, and there are such orbits around it, and the electron is somewhere in outer orbit. So when the electron jumps from an outer permitted orbit to a lower permitted orbit, it releases energy in the form of photon. Okay. So let's assume that an electron is in the nth orbit. Okay. And another electron is in the mth orbit. So their energies are En and Em. Now the electron is jumping from the nth orbit to the mth orbit. Then the frequency of radiation, let's say nu. If the nu frequency is being emitted, then the energy difference will be equal to h nu. We can say En minus Em equal to h nu. Alright. Next is if we talk about the stable state of a hydrogen, it means there is no excitation. This means what? This means the electron is in its ground state, and ground state means n equal to 1. Okay. At that time, the radius is called Bohr radius, and it is equal to R naught, which is equal to 0.529 angstrom. Alright. Next comes the time period of an electron in orbital motion. So the time taken by an electron to complete its orbital motion is called electron's time period. This is equal to T equal to 2 pi R divided by V. Okay. 2 pi R divided by V. This is the uh, time period. Any doubt here? Okay. R naught here. R naught is what? 0.529 angstrom. This is the Bohr radius. Okay. Okay, Armstrong. Armstrong means 10 to the power minus 10 meter. And here to calculate the bore's radius, we choose n equal to 1. Okay, any doubt? And if we talk about the frequency of revolution, so it is reciprocal of time period. Yes or no? So we can say this is equal to 1 upon capital T. So we can f is equal to 1 upon T. So we know that frequency is the reciprocal of time period. Now we will discuss some characteristics of an atom. Okay. So what is the orbital radius of an electron? Now R naught, which we call as Bohr's radius, that was 0.53 angstrom. So the electron is of a hydrogen atom and it is revolving in the n equal to 1 cell. Then we know that the radius of that cell. Okay, and if we want to find the radius of an electron which is revolving in a hydrogen or hydrogen like atom, then we will use the formula, that is Rn equal to 4 pi epsilon naught n square h square divided by 4 pi square z into me square, which is equal to 0.53 into n square divided by z into angstrom. Here z is the atomic number. And if we talk about the orbital velocity, so we can say orbital velocity means at what velocity the electron is revolving. So it is Vn equal to 1 upon 4 pi epsilon naught into 2 into z pi e square divided by nh. When we solving it, you will be get 2.2 into 10 to the power 6 into z by n meter per second. Okay, so here uh, 2.2 into 10 to the power 6 divided by n. This means the higher atomic number z, the higher the orbital velocity will be, and the higher cell number n. Okay, the lower the velocity will be. It means the further the electron is from the nucleus, its velocity will decrease. Then here comes orbital frequency. Basically, orbital frequency is f f equal to 1 upon capital T. So 1 upon T equal to V divided by 2 pi r, which is equal to n z square e raised to the power 4 divided by 4 epsilon naught square n cube h cube. The total energy of the orbital electron is equal to minus m into e raised to the power 4 into z square divided by 8 epsilon naught square h square n square. Okay, and we can also write equal to uh, m e raised to the power 4 divided by 8 epsilon naught square c h cube into c h z square divided by n square. So after putting the common rework constant r for other constants, so it will be equal to minus 13.6 into z square divided by n square in electron volt. So from all the formulas, we have two important terms, that is kinetic energy and potential energy. If we want to calculate the kinetic energy, so K equal to m e raised to the power 4 z square divided by 8 epsilon naught square n square h square. And if we want to calculate potential energy, so it will be minus m into e raised to the power 4 into z square divided. So the kinetic potential and total energy of the electron with r as the radius of the orbiting are as follows. Kinetic energy is equal to 1 upon 2 1 upon 4 pi epsilon naught ZE square divided by r, and the potential energy is Pe equal to minus 1 upon 4 pi epsilon naught into ZE square divided by r, and energy, or we can say total energy, is equal to minus 1 by 2 into 1 upon 4 pi epsilon naught into ZE square divided by r. Okay, so all three formula of energy, kinetic energy, potential energy and total energy, are very important for the exam. Now here one question for you. So look at here, the formula, this is the formula of the kinetic energy and this is the formula of the total energy. So here what is the common term? So let's see, please find out the common term between the kinetic energy and potential energy. So when we analyze both formula, you will be get kinetic energy and total energy. So we can say a kinetic energy, we can say total energy is equal to what? Minus of kinetic energy. Yes or no? All right. So now we will look at the relationship between these three. So first, we can say, uh, when we analyze, when it is observed that when it is observed, kinetic energy is equal to minus E, and the potential energy is equal to 2E. Okay, 2 times of total energy. This means that if we know the total energy, we can easily find the minus 13.6 into Z square by N square electron volt using this method. Okay, so once we know the total energy, we can easily find both potential energy and kinetic energy because of this relation. Now we just discussed that the radius of a hydrogen atom is proportional to n square, and the orbital velocity of the electron is also proportional to n square, and orbital velocity of the electron is also proportional to n square, but orbital velocity is inversely proportional to n, and mode of E is inversely proportional to n square. So if we talk about the difference in angular momentum of two successive orbits of a hydrogen atom, then L is delta L equal to n plus 1 h divided by 2 pi minus nh divided by 2 pi. So it will be h upon 2 pi. Clear? Angular momentum, difference in angular momentum. Students, the next topic is energy levels and hydrogen spectrum. In hydrogen spectrum, we get discrete spectral lines, not continuous, and these are categorized into 5 types of spectral lines. Okay, so out of these 5, Lyman series lies in the ultraviolet region of the spectrum, Balmer series lies in the visible region, and the remaining 3 series lie in the infrared region of spectrum. So first comes Lyman series, then Balmer, after that Paschen, then P fund. Okay, it is also written as P fund, but let's read it. Okay, so there is a 5 series, Lyman, Balmer, Paschen, bracket and P fund. Okay, actually 5 types of spectral lines are formed. So look at here. This is Lyman series. Okay. We call like n equal to 1. Okay. So this is ground state. First excited state. Second excited state, third, and so on. Okay. So when electrons goes from Lyman first state to uh, we can say ground state to excited state. Okay, and then returns back to uh, after losing energy in the ground state. So we can find their energy or energy difference. Similarly, Balmer series to Lyman series, Bastian to Balmer, bracket to Bastian, uh, P-fun to bracket, then bracket to Bastian. Okay. So the 5 spectral series of hydrogen atom are given below. First, Lyman series. Okay. In spectral lines of Lyman series, uh, Ni can be 2, 3, 4. Okay. Uh, and NF equal to 1. So we can say spectral lines of Lyman series correspond to transition of electron from higher energy levels to the ground energy level. So here final state, higher excited state is what? This one 2, 3 and 4, and ground state is 1. In that case, for Lyman series, the wave number it will be 1 upon lambda is equal to V bar is equal to R into 1 upon 1 square minus 1 upon n square. Okay. So this is the formula to calculate the wave number. So here excited states, or we can say higher energy levels, is n equal to 2, 3 and 4. Second series is Balmer series. When transition of electron take place from uh, Ni equal to 3, 4, 5 to energy level uh, from lower energy level to higher energy level. Okay. So the formula again is 1 upon lambda equal to V bar equal to R into 1 upon 2 square minus 1 upon n square. So this is the formula to calculate the wave number if spectral line series is Balmer series. Okay. Next series is Paschen series. The line of this series fall in infrared region. Okay. So in the Lyman series, in the Paschen series, the lower energy level n equal to 4, 5. Okay. N equal to 4 and excited state NF equal to 3. So 1 upon lambda equal to V bar R into 1 upon 3 square minus 1 upon n square. So where n equal to 4, 5, 6. And then bracket series. So the formula to calculate wave number in bracket series, it will be 1 upon lambda equal to V bar equal to R into 1 upon 4 square minus 1 upon n square. This is very important concept for the exam. So n equal to 5, 6, 7. So in bracket series, the electron falls on the 4th orbit and it can fall from 5th, 6th, 7th or any other higher orbit. Okay. So for this we find the wavelength using the formula. So reciprocal of wave number, we will get wavelength. Here R is the Riemann constant. All right. Next series is P-fun series. When an electron falls to the 5th orbit and falls from 6th, 7th, 8th and 9th, 10th or any other orbit, then it is called P-fun series. The formula is 1 upon lambda is equal to B bar equal to R into 1 upon 5 square minus 1 upon n square, where n equal to 6, 7, 8. Okay. So the lines obtained in this series are in the infrared region, meaning their energy is very low. Lyman series falls in the ultraviolet region, meaning we get wavelengths in ultraviolet region. In Balmer series, we get wavelengths of visible light. After that comes the bracket series, where we get wavelengths in the infrared region. And in the P-Fund series, we observe for infrared and or even lower energy wavelengths. So Ni equals 678 and to the orbit having Nf equal to 5. And energy of emitted radiation. So delta E is equal to E2 minus E1, which is equal to RCH Z square 1 upon N1 square minus 1 upon N2 square. So it will be 13.6 Z square into 1 upon N1 square minus 1 upon N2 square. And if we want to find the frequency of radiation, then we have to divide E by H. Then we will get delta E by H is equal to RCH Z square 1 upon N1 square minus 1 upon N2 square. Thank you for your attention. Now students, we will solve some practice questions based on atoms. Okay, so please wait. Hello students. Now we will solve some practice questions based on atoms. This is our disclaimer. So here we will solve some practice questions based on atoms. So first question, an, uh, an alpha particle of energy 5 mega electron volt is scattered through 180-degree by a fixed uranium nucleus. The distance of the closest approach is the order of what? So in this question, we need to find the distance of closest approach. Okay, given options: option A 1 angstrom, option B 10 to the power minus 10 centimeter, option C 10 to the power minus 12 centimeter, and option D 10 to the power minus 15 centimeter. So which option is correct? Option A, option B, option C, or option D? You have to find out the correct one. It means you have to find out the distance of the closest approach. Okay, so please solve this question. You have some time and find out the correct option. Yes, anyone? Okay, which one is correct? A, B, C, or D? you have to find out the distance of closest approach. So use the formula of distance of closest approach and put the values, and then you will be get the final answer. Okay, clear? Please solve. Anyone? Which one is correct? C, D. All right. Anyone? So the correct option is option C. Okay. Let's see the solution. So before discussing the solution, let's see the given data. Energy of alpha particle equal to 5 mega electron volt, and charge of the alpha particle Q1, let's see, is equal to 2e, and charge of the uranium nucleus Q2 equal to 92e. Okay, since uranium has 92 protons, and Coulomb constant K equal to 1 upon 4 pi epsilon naught, that is equal to 9 into 10 to the power 9 Newton meter square per Coulomb square. And we know the charge, elementary charge E, that is equal to 1.6 into 10 to the power minus 19 Coulomb. So using this information, we can find out the distance of the closest approach. So the electric potential energy at the point of the closest approach, listen carefully everyone, the electric potential energy, okay, at the point of closest approach is equal to the kinetic energy of alpha particles. So uh, this is the kinetic energy and this is the potential energy. So when we equate both, why we equate? Because at the point of closest approach both energy is equal. Now we have some given terms. Okay, so this equation can be rearranged to solve for the distance of closest approach r. So it will be 1 upon 4 pi epsilon naught into q1 q2 divided by 1 upon 2 mv square. Okay, and we know the all given terms. So just substitute the values. E equal to 1.6 into 10 to the power minus 19. So put the value of charge and other values. You will be get 9 into 10 to the power 9 into 2 into 92 into 1.6 into 10 to the power minus 19 whole square divided by 5 into 10 to the power 6 into 1.6 into 10 to the power minus 19. So when we performing calculation, r equal to 5.3 into 10 to the power minus 14 meter. Okay, so this is in meter, and if we talk about in centimeter, so it will be approximately 10 to the power minus 12 centimeter. Okay, thus the distance of the closest approach is the order of 10 to the power minus 12 centimeter, which corresponds to option C. All right, everyone, clear? Okay. Next question. To explain theory of hydrogen atom, Bohr considered. So the question asks which concept Bohr used to explain the theory of hydrogen atom. Given options: option A, quantization of linear momentum; option B, quantization of angular momentum; option C, quantization of angular frequency; and option D, quantization of energy. So which option is correct? Option A, option B, option C, or option D? Please solve this question. This is very simple question, theoretical question. Okay, so please solve and find out the answer. This is one pen down question. Which one is correct? Anyone? Yes. Which one is correct? Yes, Varsha, Karthik, VK, Shivam, Guddu, anyone? Which one is correct? All right. So the correct option is option B. Okay. So we know uh, Bohr's model of hydrogen atom is based on the quantization of angular momentum as the fundamental condition for the existence of stationary orbits. Students, this quantization ensures that the electron does not radiate energy continuously and remains in stable, non-radiating states, which is the cornerstone of Bohr's theory. Okay, good. Next question. The for the Bohr's first orbit of circumference 2 pi r, the de Broglie wavelength of the revolving electron will be what? So the question says that the circumference of the first orbit in Bohr's model is 2 pi r. Okay, it asks what the de Broglie wavelength of the electron revolving in this orbit. Okay, so there is four option and you need to find out which option correctly represents the de Broglie wavelength. Okay, so given options: option A 2 pi r, option B uh, pi r, option C 1 upon 2 pi r, and option D 1 upon 4 pi r. So which one is correct? Option A, option B, option C, or option D? Please solve this question and find out the correct option. Okay, this is very simple question. Anyone? Please solve. Yes, good. Okay, so the correct option will be option A. Okay, let's solve this question. So we know in the Bohr's model, an electron can only orbit the nucleus in specific, okay, quantized circular orbits. So the angular momentum of the electron is quantized, meaning it is an integral multiple of Planck's constant divided by 2 pi. You can say mvr equal to nh upon 2 pi, where m is the mass of the electron, v is the velocity of the electron, r is the radius of the orbit, and n is the principal quantum number. Okay. So after rearranging, we get new equation, that is 2 pi r equal to n into h upon mv, And the de Broglie wavelength is lambda equal to h upon mv. This is wavelength associated with the motion of the electron. Okay. Now we will substitute wavelength lambda into the equation. So new equation becomes, that is 2 pi r equal to n lambda. So final answer for n equal to 1. So lambda equal to 2 pi r. Okay. A option is correct. Next question. Which of the following transitions in the hydrogen atoms emits photons of highest frequency? Students, the question asks which transition in hydrogen atom emits photons of highest frequency. There are four given options, each showing a transition between two energy levels. Okay, so options are: option A, n equal to 2 to n equal to 6; option B, n equal to 6 to n equal to 2; option C, n equal to 2 to n equal to 1; and option D, n equal to 1 to n equal to 2. So which one is correct? Option A, option B, option C, or option D? So please solve this question. You have some time. Okay. After that, we will provide the solution of this question. Anyone? Please solve. Which one is correct? Okay. C, D, A. All options are correct. Okay. Anyone? So the correct option is C. So let's look at the details of the solution. So we know in a hydrogen atom, electrons exist in discrete energy levels. Okay. So when an electron transition from a higher energy level to a lower one, it emits photon. So the frequency of the emitted photon is directly related to the difference in energy between the two levels. Okay. So the energy of a photon emitted during a transition between the two levels is given by the formula, this one, RCZ square 1 upon NF square minus 1 upon Ni square, where N1 N2 are the principal, Ni is the initial orbit and NF is the final orbit. Okay. So larger the energy difference, the higher the frequency of the emitted photons. So n equal to 2 to 6, the transition from second to 6. This is an absorption process. So electron goes to higher orbit. So no photon emission. 6 to 2, emission occurs, but energy difference is smaller than ground state transition. The values of delta E is equal to 3.02 electron volt, and equal to 2 to 1 emission of ground state with energy level delta equal to 10.2 electron volt. So n equal to 1 to 2 absorption process, not emission. So we can say n equal to 2 to 1. Okay. So this one, highest energy photon, this one, from n equal to 2 to 10.2 electron volt. In the Bohr's model of the hydrogen atom, let R, V and E represents the radius of the orbit. and the speed of electron and the total energy of the electron respectively. Which of the following quantities is proportional to the quantum number n? So here we need to find out the correct options. Okay. So given options: option A E by V, option B I by E, option C VR, and option D RE. So which option is correct? Option A, option B, option C, or option D? This is capital VR. Okay, because there is a capital V. So which one is correct? Please solve this question. Anyone? Which one is correct? Please solve. A, B, C, or D? So the correct option will be option C. Let's see the solution. According to the Worth's postulate, MVR equal to NH upon 2 pi. So VR should be what? VR equal to N into H upon 2 pi M. Okay. So VR equal to N into H upon 2 pi M. So VR is directly proportional to N from this equation. Yes or no? So the product of electrons velocity and the radius of the orbit is directly proportional to principal quantum number N. Alright. Next question. The ratio of the kinetic energy to the energy of an electron in a Bohr's orbit is what? The question asks about the ratio of the kinetic energy to the total energy of an electron in a Bohr's orbit. So given options: option A -1, option B 2, option C 1:2, option D none of these. So which option is correct? Option A, option B, option C, or option D? Please solve this question. You have some time. Yes. Anyone? Which one is correct? Yes. B. A. Okay. Do you solving? All right. So the correct option is option A. So let's see the solution. we know the formula of the total energy of, uh, of an electron in a Bohr's orbit. So that is what? Total energy is equal to kinetic energy and plus potential energy. So kinetic energy is what? We can say the kinetic energy of the electron is due to its motion around the nucleus, whereas potential energy is due to the electrostatic attraction between the electron and the nucleus. It is negative and twice the magnitude of kinetic energy. So we can say P equal to 2 minus 2E into K. So substituting the expression for potential energy into the total energy equation, kinetic energy is equal to total energy is equal to kinetic energy plus minus of 2 times of kinetic energy. So when we solving it, you will get minus K. Okay. So therefore the total energy is equal to magnitude, equal in magnitude but opposite in the sign to the kinetic energy. So it will be K equal to minus E. So which one option is correct? A option. All right. Next question. If the atom FM 257 follows the Bohr's model and the radius of last orbit FM 257 is n times the Bohr's radius, then find the value of n. So the question says that atom FM 257 follows the model. It gives the radius of the last orbit is n times the Bohr's radius. Okay. So we need to find the value of n. So given options, option A 100, option B 200, option C 4 and option D 1 by 4. So which option is correct? Please solve. Which one is correct? Option A, option B, option C or option D? Yes, please solve. You have some time. You need to find the value of n. Alright. Which one is correct? Anyone? So the correct answer is D. Okay. Let's see the solution. So in Bohr's model, the radius of nth orbit is Rn equal to n square h square divided by z into me into e square. This is the formula. So where we know n is the principal quantum number and z is the atomic number, m is the mass of the electron and e is the charge of the electron. And what is h cut? That is the reduced Planck's constant. So simplified version of the formula of the radius of the last orbit. Okay. In terms of Bohr's radius, a naught equal to 0.53 angstrom. So r equal to m square divided by z into a naught. So we have a given question that the element is fermium with atomic number z equal to 100. Okay. So the isotope is 100 FM 257. So the number of protons z equal to 100. So the outermost shell is at m equal to 5 is 5. M equal to 5. The radius of the orbit n into a naught, which where we know a naught is 0.53 angstrom. So we just substitute the values. So R will be 5 square divided by 100 into A naught. Okay, so when we solving this, you will be get 1 upon 4 A naught. We can say radius of the last orbit is 1 by 4 times of the radius, Bohr's radius. Next question. Taking the Bohr's radius as A naught equal to 53 picometer, the radius of Li 2 plus ion in its ground state on the basis of Earth's model will be what? So the question gives the Bohr's radius of hydrogen as 53 picometer and asks find the radius of a lithium ion on its ground state. Okay, this is the lithium ion. Given options: option A 53 picometer, option B 27 picometer, and option D 18 picometer, and option D 13 picometer. So which option is correct? Option A, option B, option C, or option D? Please solve this question and find out the correct one. Okay, please solve. Which one is correct? Yes, I expect answer from everyone. So write the correct answer in chat box. Yes. Which one is correct? Okay. Anyone? So the correct option is option C. Okay. Let's see the solution. So we have the formula for the radius on the base of Bohr's model. So that is R equal to n square lambda square divided by 4 pi square m k into Z e square, which is equal to a naught n square divided by Z. So given lithium ion with Z equal to 3, and we know that n equal to 1 for ground state. So given that a naught is equal to 53 picometer. So substitute the value of Bohr's radius and n equal to 1 into the formula. So it will be uh, R equal to 53 into 1 square divided by 3. Okay. So after calculation, we got the values of radius as 18, approximately 18 picometer. Okay. Next question. In a hypothetical Bohr's hydrogen atom, the mass of the electron is doubled. The energy E naught and radius R naught of the first orbit will be what? Students, in this uh, question says that in a hypothetical Bohr's hydrogen atom, the mass of electron is doubled. Okay, so we need to find out the new energy E naught and radius R naught of the first orbit. Given options: option A E naught equal to minus 27.2 electron volt, R naught equal to A naught by 2. Option B E naught equal to minus 27.2 electron volt, R naught equal to A naught. Option C E naught equal to minus 13.6 electron volt, R naught equal to A naught by 2. And option D E naught equal to minus 13.6 electron volt and R naught equal to A naught. So which option is correct? Option A, option B, option C, or option D? Okay, please solve this question and find out the correct option. Anyone? Okay, please solve. Please solve. So the correct option will be option A. Let's see the solution. So we know the expression for calculating radius and energy, uh, that is Rn equal to n square h cat square divided by z k into e square into m, where m is the mass of electron, z is the atomic number, and n is the principal quantum number. Okay, h cat k e are also constants. As we know, r is inversely proportional to m. For the first orbit, n equal to 1. So therefore, r naught equal to 1 upon 2 into a naught. So En, we know energy formula, En equal to minus z square Rh divided by n square. Rh is the reverse constant, and for hydrogen atom, z equal to 1 and n equal to 1. So from this relation, we can say E is directly proportional to m. Why? Because we know the R is equal to me e raised to the power 4 divided by 8 epsilon naught square h cube into c. So new mass of the electron is doubled. So m prime equal to 2 times of m. Just put the values. So E naught equal to 2 into minus 13.6. So it will be minus 27.2 electron volt. All right. Next question. An electron makes a transition from an excited state to the ground state of hydrogen like atom or ion. So when an electron transition from an excited state to the ground state in hydrogen like atom or ion, question asked to determine how the kinetic energy, potential energy and total energy of the electron change during this transition. Okay, given options. Option A, its kinetic energy increases but potential energy and total energy decreases. Option B, kinetic energy, potential energy and total energy decreases. Option C, kinetic energy decreases, potential energy increases but the total energy remains same. Option D, kinetic energy and total energy decreases but potential energy increases. So which option is correct? Option A, B, C or D? You have to find out the correct one. Please solve this question, everyone. Which one is correct? Okay, please solve. A, B, C or D? Which one is correct? C. So A option will be correct. Okay, let's see the solution. So in hydrogen atom or hydrogen like atom such as hydrogen, lithium, helium ion, lithium ion, etc. The energy levels of electron are determined by the Bohr's model. Okay, so the energy, total energy, uh, kinetic energy and the potential energy of the electron given, uh, the formula is total energy is equal to minus 13.6 electron volt into Z square divided by N square, where Z is the atomic number and N is the principal quantum number. Alright. And the kinetic energy Ke is equal to minus 13.6 Ke equal to 13.6 electron volt into Z square divided by N square. Okay. So from this relation we can say potential energy, kinetic energy is directly proportional to Z square upon N square. And if we talk about the potential energy, the formula is Pe equal to minus 2 into 13.6 in electron volt into Z square divided by N square. Okay. So from this formula it is clear that Pe is the proportional to minus 2 Z square divided by N square. So we can say total energy is equal to kinetic energy plus potential energy. So total energy is equal to minus kinetic energy from this relation. So we can say potential energy is equal to minus 2E also from this relation. Okay. From kinetic total energy and potential energy relation. So from the equation we got that the kinetic energy of an electron is K is directly proportional to Z by N whole square. So when electron makes transition from excited state to ground state, then N decreases, so kinetic energy increases. And from the relation between these three energy, potential energy and total energy decreases. Alright. Thank you for your attention. That's all for the today lecture. Okay. In next lecture, we will talk about nuclear. Okay. Nuclear. Clear everyone? If you have any doubt, please ask. Okay. Okay. That's all. Thank you for your attention. Thank you.